Okay. Hello? Okay. Good morning, everybody. Please, we will start in a few minutes. Also, just for the people that they will present now in the first oral session, please come closer because we will start directly with the first session. So I think slowly we can start. There are a lot of people still outside. Ninon, Caroline and I, as the General Chairs of the MIDL 2024, we would like to welcome you to this event. This is, as maybe you know already, the 7th edition of MIDL, after quite some successful one that have been organizing in Europe and US. And we are very happy to see that the community actually is growing and we will fill this amphitheater here. So, of course, not very much to say about Paris. Maybe some of you, or most of you, you have visited already, or you had the opportunity to come here. It is a very beautiful city, multicultural. We hope that you will take some time to go around and to visit museums, walk around the city and the parks that they are around. The location that we are is super nice because it is in the center. So you can actually have access to a lot of parks and museums around. Jardin-des-Plantes is very close, Le Seine is very close. So you can actually, walking distance, you can find very nice environments. We also advise you not to take a car. Paris is not really designed for this. Just get a bike, walk, or even with the public transportation, it should be straightforward for you to go everywhere around. So we really hope that you will enjoy your staying here in Paris, and also you will enjoy the conference with us. So yes.
Good morning, everyone. My name is Caroline, and I have a few numbers for you regarding the number of people, the number of papers. So today we are very happy to welcome 435 registered researchers from 33 countries. The first one being France, then Germany, the Netherlands, USA, UK. This is the majority. And we had 217 papers that went through the peer review process. And finally, we have 117 papers that we are going to present during this conference. So it's an acceptance rate of 54%. This is for the numbers. We have to acknowledge the hosts. We are being here on the Sorbonne University premises. It's the Pierre et Marie campus, for those who know these names. And also we have to acknowledge Centrale Supélec, which has been a managing institution of this conference. Of course, we have our corporate sponsors that we would like to acknowledge, and especially the gold sponsors. So our gold sponsor this year are two French-based companies, Glimmer and Olea Medical, and two Germany-based companies, Infusion and Siemens Healthineers. In the bag that you were given, you can find flyers from the gold sponsors, and don't hesitate to go see Infusion that has a booth right next to the registration desk, and they can show you demos and everything. So if you are here today to show what you can do for AI and deep learning, it will also be interesting to see what this company can do for us. And so each day after the oral morning session, we will have a short presentation of companies, and it will be very interesting to show, to realize what can be done, the transfer to the clinic with all the works that you are doing. So each day there will be a short presentation of companies, and if you are PhD students, if you are postdoc, don't hesitate to go and discuss with them if you have future plans to work in the industry. And today we have two sponsor events exceptionally. You know?
Hello, so I'm Ninon. So if you manage to find like the auditorium, I guess you don't really need like the map, but know that there are two like entrances to the Sorbonne Université campus. So I guess you can just like use like the main one here, but there's also another one close to the Seine, to the river. Of course, all the orals, keynote, and sponsor events will take place here in the auditorium. The posters are spread between like the foyer of the auditorium and upstairs. So all the papers that have an oral presentation, their posters will be located in the foyer here, and all the other posters will be presented upstairs. About the oral sessions, so there's a diverse program of 9 different oral sessions. So each oral is planned for 13 minutes plus 2 minutes of questions. We cover a quite wide range of topics. So the most represented one this year was segmentation, representation learning, clinical translation, and synthesis. And so this year we have a MELBA session that includes presentations of recently accepted MELBA papers with this journal to conference track. And they will also have posters that you will be able to find upstairs. So we have 6 different poster sessions. To know where the posters are located, you have like a map and a list of like all like the posters, and of course you can just like walk around. They are ordered in the numbers like follow each other, so and the numbers you can actually find them also on the website. The posters only stay for like the day. So for all the presenters, please do remove them at the end of the day, otherwise we will have to like take them down and store them until the end of the week, and then they will disappear. So please like take down your posters at the end of the day. We have the pleasure of having 3 keynote speakers, Gaël Varocco, Laure Fournier, and Karim Lekadir. So today it will be Gaël Varocco who will present just after like the lunch break. About the breaks, so you have 2 coffee breaks per day. So the length is about like 15 minutes, but then you have all like the poster session also to enjoy like drinking coffee, tea, and eating a few things. About the lunch, so we have a 1 hour long break. You will have lunch boxes that will be available there, and then you can eat them either like upstairs and sit on these like stairs, or you can walk like to the Jardin des Plantes. It's like 5-minute walk, and you can have some fresh air and some green spaces. For like the lunch breaks, you have, and the lunch itself, you have like vegetarian option and everything. So please do check the different bags that will be available. Today we have a little like welcome reception. It will be held like here, so again in the foyer of the auditorium and upstairs. So please like stay with us at the end of the day to celebrate like the opening of MIDL 2024. Tomorrow we'll have a gala dinner. It will be at the Musée des Arts Forains, which is not far from here. It's a fun fair museum that is located in an old wine warehouse close to like the Seine. So you can either like walk there, take public transport, take a bike, but don't forget to bring like your badge tomorrow. A few more practical details about the Wi-Fi. So you can use like EduRoam, and in your tote bags you also have Wi-Fi like credentials in case you don't have EduRoam. There is a clock room with someone always like there to keep your stuff. Of course, the welcome desk is available and to like answer any like questions you might have. So please don't hesitate to talk with them. And please know that all like the orals and keynotes will be streamed on the MIDL like YouTube channel. You can, and they will be available after the conference also. So here we are 3 like welcoming you, but we are not the only one who participated to the organization of this conference. So during like the closing, we will properly thank everyone, but here you have the face of many of the people who contributed to the organization of the conference. both of like the main conference of the doctoral symposium that took place yesterday, and here you see like all the faces of the people that will help you like during the conference. So thanks to all of them. And was talking about the doctoral symposium. Actually, the conference started already a bit in advance yesterday. And so here you see like all like the PhD students who like followed some like presentation and a roundtable and visited Paris yesterday afternoon. So thanks to all like the team who organized that day. I think it was a great success. So thank you all for that. And I hand over to Alessa and Yanis.
Yeah, what a great transition. We also want to take the chance to say hi and welcome in Paris from the MIDL board. As you might know, the MIDL is not an isolated event, but it's run by the MIDL Foundation, which is governed by the MIDL board, which you can see over here. Some of us are here, and please come to us, talk to us. We're very happy to get an exchange. And I think before we get to the point we really want to make after this transition from the doctoral symposium, we have one great job, and maybe the most important job in this session, to ask for a very warm round of applause for all the local organizers that you just saw here for this great job hosting this conference in Paris. It takes a lot of effort to set all these things up, and I'm looking forward to a very smooth conference here, and especially that close to a botanical garden. I think we never had this before. So thank you very much. And as you see this board here, these are mainly people who wanted to get involved in MIDL and wanted to make our community better and think about things that make this community better and do this. And in this spirit, there's something more now. There's something new, which is the Young Researchers Board, because it is this whole community thrives from young researchers, and we also want to give them a bit more of possibility to change and shape this community.
And the idea, like Janne said, is that the young researchers represent the interests of the young researchers within the MIDL society and the foundation. We want to get you involved in the decisions about where the next conference should be, or how are the guidelines for the paper submissions, etc. And we want to hear your voice about that as well. And another aim is to stimulate the involvement of young researchers. And as some of you might already know, we have this 4 online events per year, and that is something we would like the Young Researcher Board to organize and have a strong focus on the interest. What does a young researcher need? For example, so far we had the writing workshop or reviewing workshop, and I think that are great events, but maybe you have even more ideas. What do you think? That would be great to have. That would help me in my research. And therefore, we are looking for new members. In total, this board should have 5 members, and in this year we are looking for 3. So for 3 young researchers, meaning students, PhD candidates, but also postdoc until something like 2 years after the PhD. And you would serve in that board for 2 years, and the time commitment is reasonable. It of course also depends on how much you want to do, and it will vary over time. You will get, as a board, the chances to participate in the normal board meetings, to also learn how the board makes decisions, and get a great network as well. So if you're interested, just contact Jannis and me during this event, ask questions anytime, and then send your application to Young Researcher Board at MIDL.io. But we will also send around an email later, which contains all the information and links to the website where you can do that. Looking forward to your applications.
So now we can actually get started with the first session of the day. I will just quickly show you the program for today. So soon we are the first oral session on segmentation. Then we will have a sponsor event that will take place here. Then coffee break and the first poster session. After the lunch break, you come back and we'll have the keynote by Gaël Varocco, then an oral session, another break, poster session, another oral session, sponsor events, and the welcome drinks. So now it's time for the first oral session. We have 2 chairs, so Veronica and Francesco, and 5 speakers. So I let you start the session. Thank you.
Okay, thank you very much. Good morning, everyone. Thanks to the organizers for putting together this event. And we are happy to kick off the first scientific session of today. We are your chairs. So my name is Veronica Čepligina, and I'm from the IT University of Copenhagen.
Good morning, everyone. My name is Francesco Ciompi, and I'm from Radboud University Medical Center in Nijmegen in the Netherlands.
So we will be your chairs for today and also briefly this afternoon. And in this session on segmentation, we will have 5 papers. The first one being efficiently correcting patch-based segmentation errors to control image-level performance in retinal images. And I believe this will be presented by Patrick Köhler. You have approximately 13 minutes, and we'll try to give you a heads up when you are close to time. So the floor is yours.
All right, thank you for the introduction. My name is Patrick Köhler. I'm with the Hertie Institute for AI and Brain Health at the University of Tübingen in Germany. And for this project, I was working with Jeremiah Fadukba, Philipp Behrens, as well as Lisa Koch, who is among us here right now as well, where we essentially combined a quality control framework with an error correction framework for segmentation models. More precisely, we were efficiently correcting patch-based segmentation errors to control image-level performance in retinal images. Those retinal color fundus images are an important tool to monitor and to diagnose diseases such as diabetic retinopathy, but also microvascular diseases such as hypertension, for example. Our primary objective for this project was to ensure a minimum quality for the segmentation across all images. Although we have very strong models, very strong segmentation models across the entire medical domain at the moment, even the best models may fail at segmenting individual images. However, we believe that in clinical practice, it's of utmost importance to guarantee a certain standard that is a minimum level of the segmentation quality for the efficacy of downstream tasks and for the safety and fairness. So previous approaches in those realms have focused around assessing the quality of the segmentation on an image level. That is, they try to predict the quality of the segmentation in order to defer images which were not properly segmented to a manual annotator. However, manual annotation is of course very costly, making this approach not really feasible or at least efficient on a large scale. This is exactly why we want to identify only local regions within an image that contain the major segmentation errors and defer only them to the manual annotation. Moreover, we quantify the impact that manually annotating those patches has on the overall segmentation quality of the entire image. We're working with a 5 data set which contains 800 images out of 4 different pathological subgroups, and we resized the images to 512 by 512 for our experiments. On that data, we trained a 5 ensemble of so-called full resolution units, which is a segmentation model that is specifically designed to segment thin structures such as those retinal blood vessels. It aims at retaining the full resolution of the image within its hidden representation. In parallel work led by Jeremiah Fadukba, we found that the performance of this model is actually comparable to state-of-the-art. You see an overview over the performance here on the right, where we end up with a median DI score of around 91%. So we have trained models now, which means we can take an input image and pass it through the ensemble of models, which gives us a set of segmentations. Those are probabilistic segmentations, meaning that each pixel contains a value between 0 and 1. Having multiple of those, we can aggregate them into a so-called uncertainty map. That is, we pick one pixel and we compute a measure of uncertainty, in our case the entropy, over all outputs of the individual ensemble members. We end up with a map like this, where the red regions contain pixels where the models are not so well in agreement with each other. This is exactly why we suspect that the major segmentation errors happen there, and therefore we select exactly those patches which contain the highest summed up uncertainty. For those patches, for each of them individually, we estimate how strongly the die score of the entire image would increase if we were to manually review those patches. This has the advantage that the user can basically set a threshold, a quality target, in this case 93% here on the y-axis, and can read off how many and which patches he or she has to review manually in order to guarantee this threshold. In our case, we need to review those 4 patches. Okay, let's look at each of those steps from this workflow step by step. We have a strategy now to identify those patches over the uncertainty maps, but how useful are those patches actually? In order to evaluate that, we take the prediction, and for the patches that we want to review, we insert the ground truth instead of the prediction. And afterwards, we measure how strongly the die score increases as compared to the original prediction. We look at this for the entire test set for images which were well segmented in the first place and for images which were poorly segmented in the first place. First of all, we observe that the more patches we review, the stronger the die score increases, which indicates that we're actually identifying patches that contain errors. Moreover, we observe that the images which were poorly segmented in the first place benefit much more than images which were well segmented in the first place, which is of course in alignment with our intuition, because those images contain more errors. And then if we want to quantify this effect, if we're reviewing 5 patches in each image for the entire test set, we observe a median increase in die score of around 2%, which we consider already as quite substantial, accounting for the fact that we're already at a median performance of 91% of the model. All right, so we have identified useful patches, but what we actually want to do is we want to estimate the impact that those patches have on the die score if they were to be reviewed. Here's a quick reminder of the die score. It's a measure of overlap, which requires this red area here that is the ground truth. However, we don't have the ground truth available in our scenario because we're at test time. Nevertheless, fortunately, there is an estimator which was presented at MICAI 2 years ago, which allows us to still estimate the die score without having access to this ground truth. It relies on output probabilities that are well calibrated. Unfortunately, I cannot really go into detail about the mathematics because of time, but if you're interested, make sure to pass by our poster session, here the very first poster session, and I'll be happy to explain it to you in detail how this can work. So in our framework, we use this estimator to combine the area of the image which is not the patches with the patches themselves. So for this first part here, we use the estimator, and for the second part, we simply assume for the experiments that the patches were well segmented by the manual annotator. In our case, we assume a perfect annotation, meaning a die score of 1. All right, this is how you can estimate, but how well do we estimate actually? If we're using simply the output probabilities that come out of the models after training, it doesn't work as well. But if we're actually performing a so-called temperature scaling, a post-hoc calibration method that doesn't require us to tamper with the model or anything at all, it's a very simple procedure, we observe well-behaved residuals. The left plot is for the entire images, but actually we're interested in those patches, right? So how does the estimation error behave if we're reviewing more and more patches? Well, it remains fairly constant. It doesn't change much. There is no obvious structure in the residuals. So we can use this estimator in good faith. By now, we have an entire toolbox together for a framework which allows us to set a threshold, a quality threshold, and determine for each image individually how many patches we need to review in order to satisfy this threshold. Let's simply pick 3 thresholds, in this case 88, 90, and 92% of DICE score, and compute the number of patches for each image individually, and then average over those number of patches. So, for instance, in order to satisfy this 90% threshold, we need on average to review 3.2 patches per image. All right, but what does this mean, 3.2 patches per image? Let's compare to a baseline where we fix the number of patches that we review for each image and keep it constant. And we can look at how many patches do we need in this kind of strategy to attain a comparable minimum quality. And here we actually need to correct 10 patches per image, which is even 3 times more than with our adaptive strategy. All right, so in summary, we've built a widely applicable framework because we don't make any assumptions about the architecture. And also we don't make any assumptions about the segmentation problem itself. We have applied this to retinal fundus images, but the framework applies to all kinds of segmentation problems. And this framework allows us to ensure a minimum segmentation quality by dividing manual annotation resources efficiently, meaning that for some images which are already well segmented, we don't need to invest any manual labor, but can focus the resources that we have on the images which are poorly segmented in the first place. Thank you for your attention, and I wish a nice conference to everyone.
Thank you very much for your presentation. We have some time for questions now. Take some questions from the audience. I don't know if anybody has a microphone close to them. Yeah, go ahead.
Hi, I'm Hans Meine. It looked as if you used the standard voxel-wise or pixel-wise dice for measuring segmentation quality. I'm not an expert for fundus imaging, but would centerline dice be an appropriate domain-specific measure as well? And did you also look at how much that improved? Because on slide 12, you concluded that your dice measures improved, so that that you concluded that apparently you select erroneous patches. And I wondered if you actually used the pixel-wise dice, and if you used an independent reference for measuring, for concluding this, because if you just take the same reference patches for computing the measures, maybe you overestimate the quality afterwards.
Yeah, that's a good point, thank you. The centerline dice is indeed a good measure for fundus images. We've started out with this because dice score is the obvious starting point, and it's applicable to all kinds of segmentation tasks. But you're right, we haven't reviewed the centerline dice yet, but we want to do that in future work.
And did you have independent reference for this slide?
What do you mean by independent reference?
If you take the patches, the reference patches that you use for evaluation to replace the selected patches, or if you independently label them.
So we use the patches where we have access to the ground truth from the simple training set, and we insert the ground truth from the training set for this analysis.
Okay, thank you.
I can hear you just fine, and I can maybe repeat the question.
Okay, yes, third microphone. And you mentioned the number of patches that you on average want to review per image to achieve this quality. However, in a practical setting, it would be also interesting to know for for which % of images do you not need to do anything? Do you have this number by chance?
No, actually I don't. We focused on the patches on the images where we actually need to correct something.
Thanks.
Okay, let's check. This still works, okay. So the second presentation is titled ReDefINET, Modeling Discrepancy in Tumor Segmentation Using Diffusion Models, and will be presented by the last author of the paper, Mehmet Kurt. The floor is yours.
Thank you for the introduction. Good morning, everyone. I'm Mehmet Kurt, he, him. I'm an associate professor in the departments of mechanical engineering, radiology, and neuroscience at the University of Washington. And it's a great pleasure to be here today. This is my first MIDL conference, so I'm excited to be here. So I'm going to talk about today about our work in modeling discrepancies in tumor segmentation, particularly for gliomas, by using diffusion models. So gliomas are really aggressive brain tumors, and they're the most common type of brain tumor, and they are unfortunately associated with really short median survival time. One of the best tools that we have to prolong that survival time is what we call optimal resection of the brain tumor tissue. So there's a significant clinical motivation to determine tumor boundaries based on MRIs accurately for pre-surgical planning, but also for prognostic purposes to track the progress of the tumor after surgery. And recently, as you probably are aware of, deep learning segmentation models have become really popular in this particular task, and with really increasing effectiveness and performance over the past several years. And most of you are familiar with the brain tumor segmentation challenge, BRATS, which in this field provides really high quality multi-contrast MRI data sets that have been annotated by neuroradiologists. They include ground truth masks of the glioma tumors with 3 main components that I'm going to talk about. And in the recent years, actually, the challenge has expanded to include various other types of tumors and other tasks, such as pre to post-surgical registration or in-painting tasks and whatnot. Really, I think the research community overall was able to achieve really high DICE scores. Right now, we're hovering around 90% DICE scores in this particular challenge, particularly about the glioma segmentation, and often surpassing clinical accuracy. So just to go over the training data a little bit. So the training data consists of images, modality images, multi-contrast MRI images, and ground truth segmentation masks. The 4 MRI contrasts that we have in this data set are T1, pre-contrast T1, FLAIR, T2, and contrast-enhanced T1 images. So those are the 4 contrast MRIs that we utilize in this challenge. And the 3 main tumor components that we have are the necrotic core, the enhancing tumor region around the necrotic core, and the edema region. And overall, this collection is referred to as whole tumor. The model is typically evaluated by 2 metrics, DICE similarity coefficient, DICE, as you're familiar with, and Hausdorff distance at the 95th percentile, so HD 95. recently, I think for the past 2 years, there's a new score, which is lesion-wise dice, which I'm not going to be using here just as a caveat, but I think currently the main criteria is lesion-wise dice, which also takes into account how many lesions independently you're predicting and penalizes you based on the number of lesions that you come up with. Okay, so I believe one of the most common network types that are used in this BRASS challenge has been UNets, as you might have guessed. And previous years, if you check the papers from the previous year winners, really, they are certain forms of different combinations of UNet, either vanilla UNet or some form of an optimized version, or UNets that use transformer blocks. And we essentially, or they use a combination of different strategies such as data augmentation, ensemble methods, or various post-processing methods to improve the segmentation quality. So our motivation with this study was to test the premise of denoising diffusion models in tumor segmentation. Diffusion models are really better at mode coverage as compared to GANs and potentially capturing the high variability and irregularity of the tumor boundaries and generating high quality images accordingly. And to compare the performance of our diffusion model, I want to first introduce our baseline model, which is an optimized unit adapted actually from 2021's BRASS challenge winner. So we are using that as our baseline model, and we are improving upon that model by using denoising diffusion models. All right, so I want to go over some of the previous kind of attempts to use diffusion models particularly for BRASS challenge. So one of the papers that we were able to identify is called DiffUNet, which essentially uses a denoising UNet and conditions the model by using the modality conditioning from the MRI images for MRI contrasts, and in the inference stage, segmentation ensembles are formed by using multiple noise images. And this is one of the models that we will test and compare against our model, just as a caveat. So some of the other models that we have come up with to compare against the model that I'm going to introduce is essentially taking this DiffUNet model, but instead of conditioning it based on the 4 MRI contrast images, we are going to condition it based on the baseline UNet prediction. What is that? So we use the optimized UNet from previous BRASS year challenge, and we condition this diffusion model with the baseline UNet prediction from that optimized UNet model. That's the first model that we're going to test our model against. So we call that UNet augmented diffusion. The second variation of this will be adding also the MRIs, the 4 multi-contrast MRIs as a conditioning input onto the baseline UNet prediction. So this will be the second variation of this model that we're going to test. Our model, we call RedefineNet, is really similar actually to this, what I'm modeling here, with just a minor difference. Instead of trying to predict the segmentation masks, what we're going to try to do is we're going to try to predict the discrepancy between the UNet output from the optimized UNet and the ground truth mask. So essentially, we're trying to model the discrepancy between the optimized UNet and the ground truth mask, and that's the output. And when you combine the prediction from our model with the UNet model, ideally you should get the ground truth mask, right? So this is the model that we're going to test, and we call that RedefineNet. And for discrepancy modeling here, we're using a denoising diffusion, but we ask the question, is this fair? Because the whole premise of this study is to test the premise of using a diffusion model, but we are also doing discrepancy modeling here. So to check this can be indeed attributed to the diffusion model, we also used another optimized UNet, separate, different UNet from the baseline UNet model, to model this discrepancy instead of a diffusion model. Essentially, that's the same, right? The idea is the same. We use the multi-contrast MRI and the baseline UNet prediction, but instead of modeling the discrepancies between the initial prediction and the ground truth mask, with a diffusion model, we model it with another UNet. So this is another model that we're going to test our performance against. We call this discrepancy UNet. So I went over a lot of different models, but I'm just going to now show you an overall comparison. This is from single fold, but I'm going to show multi-fold results as well. So just to test the premise of all these models, we compared them for the whole tumor region, enhancing tumor region, and the tumor core. So as you might have guessed, of course, the baseline UNET model performs quite well already. I'm presenting the results for DICE and HD95 here. Some observations is that first thing to look at is that the diff UNET model underperforms, the initial diffusion model that has been proposed underperforms compared to the baseline UNET model. That's the first observation that I'm going to have. The second observation is that when we compare modeling discrepancies with the diffusion model, which is the redefined model, compared to modeling discrepancies with a UNET, we see a substantial change, especially in the HT score with respect to the discrepancy unit. So this is really promising from our study perspective. And if you look at UNET augmented diffusion models, UA diffusion models, one of which is just using the UNET outputs, the other one is UNET outputs and the images, conditioning based on images and UNET outputs are better, yield better performance, both in DICE and HT, compared to just conditioning on the UNET outputs. And these results, so we took the 4 most promising models here and did a 5-fold cross-validation. I'm going to show that next. And the results across different folds are quite consistent. And consistently what we observe is that redefined yields comparable performance in DICE compared to other models. However, achieves substantial improvement in the HT scores, especially in the ET region. And this is, I think, something that we were excited by, because enhancing tumor typically has more irregular boundaries that are associated with higher spatial frequency features. And we would expect, based on our initial hypothesis, for diffusion models actually to improve, and HT being a boundary score, we would expect our diffusion model to actually improve the enhancing tumor scores better than the other regions. And this is indeed what we see, particularly in the HD95 score. So we were particularly excited and encouraged by this. So to summarize, in this research, we proposed a tumor segmentation framework called redefined, which modeled discrepancies based on a baseline UNET model and a ground truth mask. Our code is available on our GitHub page if you want to test it out. This segmentation framework achieved a substantial improvement in the HD95 score compared to other networks that we have tried. And we hope that this is kind of an initial study to test the benefit of using denoising diffusion models in brain tumor segmentation. So with that, I would like to thank my research group, my postdoc, Tiani, and my graduate students, and our collaborator, neurosurgery collaborator, Jake Ruzovic from University of Washington Neurologic Surgery, and I'm happy to entertain any questions. Thank you so much.
Thanks, Mehmet, for your presentation. We have a few minutes for questions. Any question? Yeah, there is one over there.
Yeah.
Did you consider an ensembling, or is that included? I didn't.
Yes, it's included. Yeah.
How many is a good number there?
I don't remember at the top of my head, but I can get back to you with an answer later. Yeah.
5 or more?
Yeah, I think more.
Okay. Yeah. Right. Thanks.
There's another question over there.
Here? Again, most of the details are in the paper, but I'm happy to get back to you on that. So I don't remember off the top of my head what was the function, but I'm happy to get back to you.
Yeah, there was already a question over there.
Thank you. A question is regarding the denoising unit. Could you, if I got it correctly, so you will have to predict n times the denoising unit at prediction to come forward to a segmentation. Can you maybe comment on the prediction time of the workflow compared to just applying the unit once in the original method?
Your prediction time, like computation time?
Yes.
Yes, this is one of the challenges with these models. It is indeed, it takes, we trained on 2 GPUs for one fold. It takes about 4 to 5 days. Yeah.
I mean more in the at deployment in the clinic, for instance. So
I cannot hear you.
I mean in deployment in the clinic. So what is the prediction time if you just use a regular unit versus you use a denoising unit where I guess you will have to have a couple of iterations before coming from the
once
noise
it's
to the
pre-trained,
segmentation.
it shouldn't be an issue, right? So I think it would have comparable prediction time.
Yes, thanks.
Yeah, thank you.
Okay, there is maybe time for one last burning question. Very short one over here, maybe.
Oh, sorry. Well, thank you. Have you ever applied about if we use another prediction of the input model, input mask? That is, we use a different model, or use the ground truth mask as the input.
Use what is input? Sorry.
In the left top, left top, the baseline UNet prediction UI.
Yeah.
Have you applied about if we use a model that not that good enough to predict the the mask?
That's an excellent question. Thank you for asking that. Yes, we have tried that. We have tried actually other forms of, so we have tried a vanilla unit, not an optimized unit. We have seen actually more significant improvements by using the redefined nets. So it seems, based on just an initial hypothesis, it seems like if your prediction is worse, actually the improvement that you can achieve with the redefining process is better. So yes, we have tested a few other networks that are not in the paper, but I'm happy to talk to you in person about that. Thank you.
Okay, let's thank our speaker again.
Thank you.
Very space on the first rows for the people who are standing upstairs, and please wait for the mic for the questions. I mean, we have 2 people running to you. Please wait for them.
All right. Yeah, thank you very much for attending my talk. I'm Elias Baumann, and I'm from the University of Bern in Switzerland. And today I'd like to present HAVA-Next, a fast nuclei segmentation and classification pipeline for next-generation histopathology. And I want to start out by showing you a hematoxylin and eosin-stained colorectal cancer section that is scanned. And actually, this is what we look at in pathology a lot. And most of what you see here is cancer. So it's all cancer tissue. And if we take a region of interest here from this slide and we zoom in, we actually see tissue composition. And on the top left here, you can see cancer sort of invading into the tissue. And on the bottom right, you can see immune response. And this is really interesting for the prognosis of the patient because these are essentially the features of the disease that are represented on the image. Yeah, exactly. So what we want to do in histopathology, and especially in this context, is actually to quantify the cellular composition of the disease. We want to investigate spatial relationships between each of the cells of interest, and we want to analyze the cellular morphology. And finally, obviously, you want to correlate results with disease features. And if we apply deep learning, we can actually do that, and nuclei segmentation and classification allows us to extract exactly this information from already routine diagnostic slides. So this is something that can be deployed readily in the clinic. So I think this already kind of makes clear where the motivation is coming from. Essentially, the cellular composition of, for example, cancer, but also other diseases, really impacts the disease outcome. But existing nuclei segmentation and classification pipelines and algorithms are really slow, so it takes a lot of time for them to run, multiple hours per slide, and that's not feasible. And finally, a lot of the publicly available datasets actually lack interesting labels. So there's more cell types available in the tissue that we can identify on H&E, but oftentimes they're missing. So to solve all of these problems, we're introducing HoverNext. And this is what it looks like. So it's essentially a very straightforward pipeline. We have an H&E whole slide image. We have to tile it because it's very large. We feed it into a deep learning model, which is HoverNext. Then there is a cache to intermediately store the results. There's a stitcher to recombine all of the tiles together. And then finally, we have a whole slide output. The model itself has actually already been published before. We submitted this to the Conic 2022 challenge and placed second in that challenge. But one of the big challenges there was actually our model was the slowest of all of the submissions. You had a 1 hour time frame to submit your results. And our model took, I think, 58 minutes of that 1 hour. And all of the other models were way faster, like the 10 to 15 minutes. So we had to do a lot of improvements to actually translate our model to and deploy it in the clinic. So if you're familiar with HoverNet, which is another published nuclei segmentation and classification pipeline, the architecture is actually quite similar. So we have an encoder-decoder model. There's 2 branches. HoverNet actually has 3 branches. So what we are doing is we are merging the instance decoder into essentially like one branch where we predict horizontal and vertical center point vectors. And we are also predicting a 3-class border, center, and background map. And we are basically combining that in one decoder. And we are using the 3-class border, center, and background map also directly to use it for watershed to get the instance segmentation. Also for the encoder, we're using ConvNext, that's where the name is coming from, and we are publishing models with the large base and tiny size. The other important thing of this pipeline is actually the stitcher. So one of the important parts is obviously when you run a model on tiles, you need to combine the tiles back together, and there's a bunch of issues with that, and for example in HoverNet, that's quite slow. So the big problem is that resolving per tile overlaps, because you're going to have some overlap to do the instance merging correctly, is really expensive. And the overlap still needs to be large enough so you can avoid false splits. So what's the solution? Well, you don't post-process directly. You essentially stitch the raw output of the model together, and then you post-process. And then you only have to solve overlaps between really large areas. Well, you can essentially just do the entire whole slide image at once, but then you need a lot of memory. So what we did is essentially just tile it into large regions and then resolve the overlap in those large regions. And the nice thing about doing that actually on large regions is that you can still parallelize watershed, because otherwise you'd have to run watershed on the entire whole slide image, and that would take a lot of time. So the other interesting thing, and we were actually really interested in including mitotic figures into our colorectal cancer evaluation. And the lizard dataset, which is publicly available, doesn't actually have that annotation. So what we did is we actually took H&E whole slide images and we restained them. So we essentially washed the color away and we applied a PHE3 antibody via immunohistochemistry, and you see that in brown. The highlighted mitotic figures, and then we registered those images with simple elastics, and we applied our only on lizard trained model on those images, and then could essentially take the instance segmentation as sort of annotated regions, and whenever there is a IHC positive area, we just take that information to generate a ground truth. And whenever we have something in the PHE3 that is not detected as an instance, we can also just add that. However, the problem then is, what about the other classes? So all of the other labels are still missing. And so this is where we actually used SC++, which is a self-training routine that has been previously published. And how that works is, essentially, you're training the model on your fake labeled or pseudo-labeled data set and on the already labeled data set together. And then first you're selecting easy samples. So you're basically finding easy samples, you're training on those easy samples. Then you're finding hard samples, you train together with hard and easy samples. And while you're doing that, you're always relabeling the sort of pseudo-labeled data set. And finally, when you're done with that process, you train on the entire combined data sets and you produce the final models. And what is an easy sample and what is a hard sample? The idea is actually quite simple. So an easy sample is something that is very consistent across training. So if you take an early checkpoint and you take a late checkpoint, the output that the model produces should be really similar. That means it's easy because it was learned early on in the training. And a hard sample is one that, well, it's the opposite. So it's one that wasn't learned early on, so there's a really high discrepancy between the early checkpoint of model training and late checkpoint. And what is also important is that we obviously also are predicting mitosis labels on the lizard dataset, because there they also don't exist. So for our analysis, we're using two publicly available datasets, the PanUke dataset and the lizard dataset. The PanUke dataset has 5 cell types, neoplastic, non-neoplastic, epithelium, inflammatory cells, connective, and dead. And the lizard dataset has a more heavy focus on immune cells and a blanket catch-all connective tissue class. And then we are publishing 4 new datasets with this, the mitosis dataset, which contains the ground truth mitosis annotations and also predicted lizard cell types. Then we have the mitosis annotations for lizards, so you can basically overlay those on lizard and use that additional class for training. Then we have a holdout eosinophil validation set where we manually annotated eosinophils because we are curious on the eosinophil prediction performance of the model. And we have another holdout mitosis validation set where we had 3 readers that manually annotated mitosis on an H&E directly. So what are the results? The first comparison that we did was actually with our own model that we submitted to the challenge, because we are obviously curious. We had a very slow model, we sped it up, so how much worse were we? And we're happy to say that we are still the same performance, so that's really good. And you can see that in terms of panoptic quality, which was back then the metric that has been used during challenge evaluation, the large HARVANEX is actually on par as the conic submission. However, when we actually investigate a bit more, we see that the F1 score and Hausdorff distance sort of tell different stories, where the model comparison is flipped in F1 score compared to Hausdorff distance, where HARVANEX is actually better in F1 score and worse in Hausdorff distance. And one reason for that is actually in the original challenge, and that's partially why it was so slow, we used the convex hull-based filtering. So we produced convex hulls for each of the nuclei segmentations, so for each individual nucleus, to figure out if it was a nice and round nucleus. So if it wasn't, we kicked that out. But obviously that's not really what you want in a real setting. What I can also highlight here is that there is a nice publication on why you shouldn't use panoptic quality as an evaluation metric in nuclei segmentation. So we included other evaluation metrics in the paper as well. What is also nice to see is that predicting mitosis seems to work really well as well. So the F1 score is on par with other rare classes, and mitosis are also really rare in this data set. And what was also interesting to see is that actually the model is quite good at predicting eosinophils in our holdout test set, and way better than on the lizard data set. What we also did is we are employing test time augmentations. So we obviously had to compare what is a good amount of test time augmentations. And what we saw is that, well, obviously if you have more, there's a tendency that the results will improve. But already at 4 test time augmentations, you get a nice bump. And then if you think about the diminishing returns and that you want to also still have a fast model, like using 4 test time augmentations seems to be the best choice. What we also observed is that particularly for the rare classes, the test time augmentations seem to help more in improving model results. Then we also had to compare obviously on PanUke, which is the sort of benchmark data set for all of the other nuclei segmentation and classification data sets. And what we found is that our model performs competitively, but is not as good as the cell VIT with the SAM backbone, but at least it's better than HARVANET. However, when we actually looked on this on a qualitative side, we actually looked at whole slide images, we checked out what are the results. We saw some interesting things where in this lymph node in the top row, you can see in red is basically tumor epithelium predictions, that for cell VIT, there's these artifact tiles where you essentially have these tile-based outputs where everything is predicted as the same class. And there seems to be some kind of normalization issue so that you can't actually deploy this anywhere because you don't have these square tumor areas anywhere, right? And HARVANEX actually does quite a good job in that region. if you look at the bottom row, you actually have a normal epithelial mucosa. So there, everything should be green in theory. None of the models actually do this. So none of the models actually predict this correctly. However, HoverNext does the best job at finding those green dots in this area, so finding non-neoplastic epithelium there. And the last and most important thing, I guess, is that HoverNext is actually super fast. So it's 5 times faster than CellVIT. It's 17 times faster than HoverNet. And what does that actually mean? So essentially, if you have a resection whole slide image with quite the large specimen on there, you are talking about 11 to 27 minutes for one slide. And if you have a biopsy, so like a smaller tissue object, it's like 2 to 8 minutes. And this is for the PanUke trained model at 0.25 MPP, so that's the sort of highest resolution. So you can halve the time if you're using the lizard trained model because it runs at 0.5 microns per pixel.
You have about a minute left.
Yeah, I'm going to skip that, and I'm going to go straight to the conclusions. If you have questions on how to get there, basically, we provide a competitive nuclei segmentation and classification pipeline. We extend the publicly available lizard dataset with mitosis and publish further validation data. And HoverNext is highly efficient and optimized for large-scale investigations. With that, I would like to thank all of my collaborators, the people at the Odyssey Digital Pathology Bern Group and the Institute for Tissue Medicine and Pathology of the University of Bern. So thank you very much.
We have time for some questions. And please do wait for the mic if you are asking a question. Somewhere in the back there, or? Okay, so maybe, oh yes, here in the front left. Yeah.
Thank you, very interesting talk, and nice to see that it runs now this fast. At the beginning you were mentioning that this is important to connect this cell quantification with some endpoint clinical tasks, but I didn't see any of those results there.
Yeah, so you have to wait for my next publication for that.
Okay, all right.
Any other questions? Okay, so there's something also I wanted to ask. So as I understand, you're combining things from different data sets into the same data set. Is there like a danger there that due to some slight domain shift, you might be predicting more like things, which data set is it from rather than what class it is?
Yeah, so that's why we actually added the extra test set to sort of check if that's true. And also, again, we validated on the same data set, so the same test set that we had before, to make sure that we are not decreasing performance because suddenly we are learning something that we shouldn't learn. But obviously, like if you're doing this sort of self-training routine, you're gonna maybe learn something that is wrong because it was already wrong on the first data set.
Thank you. So I think in the interest of time, we move on. Let's thank the speaker again.
Okay, the title of the next presentation is Semi-Supervised Segmentation via Embedding Matching, and will be presented by Wei Xie. Xie, the floor is yours.
Yeah, thank you, Francesco. Hello, everyone. I'm happy to present our work on semi-supervised segmentation using this embedding matching approach. We are from the Striker AI Research Group. My name is Wei Xie, and I'm presenting on behalf of my other colleagues. I would like to spend a couple of seconds to talk about the semi-supervised segmentation. What is the motivation? It's pretty simple. So we have the data annotation scarcity issue, especially for the 3D medical images such as CT scans. So the question we want to answer is that can we only use a small number of labeled cases and leveraging those large number of CT scans be available from the PAC systems for training a good segmentation model? I think there is still additional information available in those CT images, although it's not labeled. For instance, they're all from the different, for instance, could be from different scanners, could have a different patient population, or they might have different anatomical and pathological features. So how we can leverage them? That's the question for a semi-supervised segmentation. The standard annotation for this topic is basically we have two different data set. L is the labeled images, U as unlabeled images. Usually L is much smaller than U. Commonly there are two types of approach. One is called pseudo labeling, which is also very easy. So you can train a model based on the labeled data set, then you try to pseudo label the other unlabeled images, and then you come up with a combination of the data set, and you could retrain a model on top of that. And the key idea is that you have to do some kind of model or data expansion or jittering so that you don't end up with the same model. So let's say you train a model A on the data set L, and you use that to label the data set U, then you do L plus U training for the same model. Then in my opinion, that will not give you very much improvement based on the same networks. So what people do is that they add some modifications, either in the input or in the network itself, or even in the intermediate feature maps. So that's also the idea of the consistent training. So kind of with the same input, if you add modifications to different version modifications, do you end up with the same, you want to force the training so that the output will be close to each other. Also another kind of a regularization techniques. In this field, there's a famous paradigms called student and teacher. Again, semi-supervised way is one way to deal with the kind of data scarcity issue. The self-supervised way, they also use this idea a lot. In this student-teacher work, so basically they have two network with the same architecture. The student network is trained with the standard backpropagation, but the teacher is basically created by the moving average of a student. You can think about it as a sample or aggregate version of the student knowledge. Then through consistent training, they actually ensure that, okay, with the same input, apply a different kind of random jittering, adding noise, do you get a similar output or not? So that's the way they supervise the training on the unlabeled data set. Later on, there is another work called uncertainty aware teacher or mean teacher. There, the author says that, okay, the teacher's output sometimes are misleading, so they apply some uncertainty analysis to figure out which regions are actually in a teacher's output where the uncertainty is low. They use that as a pseudo label, and they completely discarded the rest of the regions for training, for consistent training. But there is a problem because you actually ignore lots of the voxels. In medical images, those regions are with high uncertainty, mostly on the object boundaries. You want to think about a way actually how to include those voxels in your pseudo labeling. There's lots of other ways. So our approach is basically attacking these issues. The first part of our work is basically same as uncertainty aware mean teacher. So you can see that the teacher network is using unlabeled image as the input and do uncertainty analysis, whether whether it's a certain, then we use that as pseudo label to train the student. But what we add is that this conditional block. What if the uncertain analysis says that this is not good enough? Can we think about a way to do that? So our way is essentially use nearest neighbor classification. So in each mini batch, we have a number of labeled cases. We have a number of unlabeled cases. When we're dealing with those unlabeled cases, we actually borrow the labels from the labeled images. We actually use teacher network to take the features, and we can actually sample, let's say, k number of object embeddings or a key number of background embeddings, because we know we have the label, we know which region is the object, which are the background. Then we take the features from the unlabeled images. We could do this kind of pairwise, sorry for that, pairwise similarity measures using some kernels. So in our work, we do the Gaussian similarity, show that that's the best one. And then you can come up with these similarity maps. That's these two things with weird coloring schema in the middle in this diagram. And you can actually compute argmax, then you get a pseudo labels directly, so you can use that as a pseudo label to train your student network. Or you can also do this entropy minimization. It's a way to actually train the nearest neighbor classifiers to make sure that the object similarity maps are far from the background similarity maps. So that's pushing the decision boundary of the nearest neighbor classification. So yeah, this is a conclusion for the rationale of this approach. So we use label propagation through the samples of set of KN classifiers using the random selection of the neighboring sampling from the labeled voxels. And we also do this, okay, there's something wrong here. Let me go back to the presentation mode. I don't have a mouse.
Indeed.
Maybe F5. I remember the shortcut here, so I'm not sure. Okay. So I will go to the data set. So in total, we have 950 CT scans subjects. 350 for training, 100 validation, 500 tests. We do training evaluation only on the 3D patch level, so for simplicity. And in the end, we sampled 6,200 something for training, 1,000 something for validation, and 1,200 for test. And we also actually measure the segmentation complexity for each patch by actually evaluating the surface deviation from the common shapes, from the common anatomy using statistical shape models. And that's this SDS score means. So we do the balance sampling for the test set, make sure all the kind of complicated cases or easy cases are well distributed in our test set. The final results actually shows that our approach with only 50 3D patches sampled out of 3 CT scans, I think, will give you 3.3 millimeter Hausdorff distance with a 95% percentile. That's best compared to other performance. But the other side also shows that for all of this approach, if you want to get less than 2 millimeter Hausdorff distance, which would be a fairly good segmentation model, you need at least have 1,000 3D patches. So the conclusion will be that even the semi-supervised approach with a small number of cases works well, but if you want to really achieve good performance, you need to still have a fair amount of 3D patches labeled. I think that reason is because that if you, depending on how, like the variation in your data set, if you really like the lots of pathological or anatomical changes, you need to have at least some cases to cover the edge cases. Same as we show in the qualitative study, you see this middle row with meta artifact because we don't have lots of cases covered, and there are still lots of inconsistency in the final results. The summary of our approach is basically the key innovation is that we use the embedding correspondence to transfer labels from the labeled voxels to unlabeled ones. You can think about this approach as a way we do model expansion. We use a different non-parametric model. If you think about how student-teacher works, that they have a teacher basically is a different version of the student. So you need to have this kind of difference in your architecture design for semi-supervised learning. We also choose a different sampling strategy to introduce the noise. So we do random sampling for those labeled voxels for the entropy minimization or the neighboring matching loss computation. So in this way, we think we can leverage that the unreliable regions from the teacher output, we can label them in a different approach, and that will improve the overall performance. And thanks for your attention. I'm happy to take any questions.
Thanks, Chair, for your presentation. We have quite some time for questions. Is there any question from the audience? No, maybe I can start.
Yeah.
So could you go back to the slide where you have the table with the results?
Yeah.
Okay. Yeah, I was missing that because something that I read in your paper, at some point you did an ablation study to see the contribution of the different losses that you have. And essentially at some point you have one loss term which relies essentially on finding the nearest neighbor. And you saw that actually getting rid of that term doesn't decrease the performance that much. I think maybe if you can go back to the figure with the terms. Yeah, the neighbor matching loss. Yeah. I was wondering about the contribution of that term.
Yeah, that's basically when we use the classifier to get the pseudo labels, how much performance gain from that. I think the reason is because we only apply this loss for the region where the uncertainty is high. And those regions are actually a small part of the entire, let's say, volume. And we see improvement, but not much, I think, because of the regions are generally small. But I think if you look at Hausdorff distance, there's a bigger change than the LU. I think that's my explanation. Hope that answers questions. Yeah. The other part of our work is this intrabeam minimization, helps you to push the features like more discriminative to separate the object and the background. I think that one has a bigger contribution from the ablation.
All right. Yeah. Thanks.
Thank you.
There are questions? Yeah, one over there. Maybe wait for a microphone.
Well, thank you. I have some questions about the result table and why the fully supervised model have lower performance than the below semi-supervised models. I think it's not very...
You mean the first row, right?
Yeah, the supervised baseline is much lower than the below lines.
It's because we only use 50 street patches in training the supervised baseline. So the column-wise, the first column, first row is that the performance you get with only 50 images in training for the supervised baseline. Does this answer the questions?
Yeah. The amount of images in the dataset is 6,000 about.
So in total, we have 6,266 3D patches in training. That's the last columns here. You see that's the final, if you include all the data available, then you get this performance. It's still better than any semi-supervised approach.
Yeah, I know. Thank you.
Yeah, thank you. Happy to take any questions in the poster as well. So probably.
Yep. Thanks. Also, in the interest of time, let's move on to the next presentation. Let's thank Chi again.
Yeah. Thank you.
Okay, and for the last paper in this session, we have boundary-aware contrastive learning for semi-supervised nuclei instance segmentation, and it will be presented by Zhang Ye.
Hello everyone, I'm Zhang Ye from Harbin Institute of Technology. Today, I am so excited here to share my work, Boundary-Aware Contrastive Learning for Semi-Supervised Nuclei Instance Segmentation. And nuclei segmentation is very important in pathological image analysis. First, cell is a basic component of pathological image. And second, accurate nuclei segmentation can provide support for the tumor microenvironment analysis. For example, if we get a accurate segmentation result, we can build a cell graph and to achieve the cancer diagnosis and the patient pro-diagnosis. But the pathological image have evident difference with the traditional medical image. Here we give example. From the left picture, we can see the medical image usually is small and its size is around 500 by 500. But for a pathological image, its size may be 500,000 by 400,000. So if we train a nuclear segmentation model, it's very difficult to label all nuclei. Here we give a nuclear segmentation process. For example, first, some people need to collaborative annotation on nuclei, and it may be need some pathologists and some students. And second, it also need an iteratively check process and to correct some incorrectly labeled nuclei. So it's imperative to train a semi-supervised nuclear instance segmentation model, and the pathologists just need to label part region of the WSI, and with the labeled data and the most unlabeled data, we can train a semi-supervised model. But nowadays, the semi-supervised nuclear segmentation model still face some key challenges. First, the nuclear morphology is varying. Here we give some examples, such as the fibrocyte, lymphocyte, and metastatic cell, and its shape is varying, such as spiral, circle, and irregular. Besides, the nuclear boundary is blurry. Maybe it's caused by the HE staining process. So we propose to solve the boundary blurring problem. Here we train a teacher-student module. And first, our method consists of two key modules, and one is low-resolution denoising module, and another is cross-hour contrastive learning module. First is a low-resolution denoising module. From the left picture, we give an example of the nuclear prediction probability output. We can see its boundary is a cause. To smooth the boundary, maybe we can use the downsampling operation, but it will lose some detailed information, yes. So how to solve this problem? We can cut the original high-resolution map and the low-resolution map to achieve nuclear segmentation, like this. Yes. First, the original prediction map be downsampled to a low resolution, and we calculate two losses, naive mask head and the low resolution denoising head. So by this way, we can simultaneously employ the naive mask head and low resolution denoising head. And next is the cross-hour contrasting learning module. Similarly, we give an example of the probability output. From the left picture, we can see the center probability output is following a trend. From the nuclear center to the boundary, its probability is decreased. And in the nuclear center, its probability is 0.98, but in the boundary, its probability is 0.46. So we may propose to enhance the boundary discrimination, and we use the three types of method. First, we can find the nuclear boundary, like the green part. And we use some deletion and erosion operations, and we find the outer boundary and the inner boundary, like the labeled, yes. And use the true boundary, outer boundary, and the inner boundary. We can split the entire LI into the 4 regions. And sorry, this some mistake. I don't know why. The four reasons, for example, the orange response to the nuclear region, and the red corresponds to the background, and the green and the purple correspond to the hard samples. And we just need to enhance this part feature discrimination ability. And so we use a simple operation, and we set the sample ratio is alpha to sample from the full region. Like this. Okay. Why this? Okay. For example, for the foreground, we average all foreground feature and get the QF, and average all the background pixel feature get the QB, and we calculate 4 couples of losses like this. For example, QF, KB, and KF. By the contrastive learning calculation, we can push the QB and KB together, and QB, KF away. Similarly, QB, KO together, and QB, KI away. And by this method, we can enhance the boundary feature discrimination ability. And the following is the contrastive learning calculation equation. And our whole training use the mask RCN as our baseline. It's a 2-stage training method. It consists of detection head and segmentation head. And our whole framework is a teacher-student network. First, we train a teacher net, use a label data like this, and the training loss is segmentation loss and detection loss. Next, we use the well-trained teacher module to generate procedure label for the unlabeled data like this, and it's just an inference method. And third, we can test the label data and the procedure label data to train a student module. In the student module, we use the above designed modules, low resolution denoising module, and cross-RI contrastive learning module. Yeah. And this is our experiments, and from the table we can see our personal method bus have an evident improvement compared to the MMT-PSM, in depth about 5% improvement, and also it have evident improvement compared with the supervised method, mask RCN. Okay. Next is a visualization comparison with result. From the result, we can see our method can detect more nuclei like this, and our method can solve some staying cost problem like this region. Yes. Okay. Next is our ablation studies, and we set different distance D and different sample ratio alpha. From the D, we can see whatever the D is small or large, it will lead to the performance decrease. And with the alpha increase, the performance is also increased, but it will enhance the calculation effort, so we choose 0.5. Yes, and this our conclusion. In the future, we will co-training the teacher and student net to enhance the feature interaction. And this our acknowledgement and our team. And in the end, advertisement for myself. If any group need a postdoctor, please contact me. And welcome to our poster. Thank you.
Thank you very much for the presentation. So we have some time for questions. Yes.
Thank you for the really good presentation. So I just wondering, did you just achieve the nuclei instance level segmentation or the semantic? Including the nuclei classification as well?
Pardon, please slowly speak.
Because you're comparing your result in the PQ score, and I'm wondering, is that only the instance level segmentation or the segmentation including the nuclei classes as well?
We don't have class, just instance segmentation.
Okay, and another question is, you proposed a low resolution denoising hat mechanism to finding the best boundary for the nuclei. So I'm wondering, in the paper, I have noticed that when you're applying more data sets, like a half of the data set for training, but you got a little bit lower in the DICE score. Can you explain a little bit?
I guess you means is the boundary maybe influence the performance? Yeah?
Yeah, so...
yes, true, the boundary will influence, but when we train the low resolution, sorry, this part, in fact, we train this branch, we use a feature from the low resolution and contrastive feature, so it can ease this problem, I think.
Thank you.
Are there any other questions? So, yeah, in the back, please.
Hello, thank you for the presentation. So for your cross ROI contrastive learning part, you divide this region into the nuclei, the inner bound sort of, and the outer bound.
Yeah.
And you sample some points from each of those. Can you please repeat how you calculate the contrastive learning loss from those?
Okay,
exactly.
First, we average foreground pixel feature gets the foreground representation, and average background feature gets the background representation as query. And in this sample result, we want to push the same class near and different class away. So we just calculate the contrastive learning like this. For example, in this blue corresponds to background, and this is also background. So by this way, background and background close, and this KF is the foreground, and it will let the background and foreground away. Sorry, then this and this is same. Yeah.
Okay, great. No, that makes sense. Thank you.
Okay. If there's a tiny last question. And if not, let's thank the speaker again. And we conclude this session, and I'll hand back over to the organizers.
Okay, before moving to the sponsor event, we have a short announcement to make. So if you have a job offer you would like to publish, then you can have it published on the MIDL website. So please like contact the webmaster at MIDL.io, and it will be like published on the job board on the MIDL website. So no sponsor event. I lost my, yes, you're there.
Thank you, Nino. Okay, so we can start with the first sponsor event, and we are very happy to have one of our gold sponsor, Glimmer, and in particular Gabrielle Misrahi, the head of the AI team of in Glimmer, will give a presentation of the company.
All right, thank you for having me. So as you said, I'm Gabriel Misraki. I work for Glimmer, and I head the AI department there. And I want to give you a brief presentation of what Glimmer does and give you some hints as to what our research is. I think that could maybe interest you. So see beyond with AI, that's our motto. It illustrates what our mission is, get the radiologist and the healthcare industry in general more out of their medical images. And we've started that mission by building our first set of products that we coined the AI Copilot for radiologists. We started off with X-ray products, and the flagship of which is BoneView, which is our product for fracture detection and along other pathologies as well. We also detect dislocations, articular effusions, and bone lesions. We then moved on to lung pathologies with ChestView that does the same kind of job, but for 5 acute lung pathologies. Then came Bone Metrics, which is a very efficient product for radiologists in the sense that it makes them, it gains them a lot of time by automating around 100 measurements that they have to commonly make on MSK X-rays. A good example of which is the Cobb angle, which you have to measure the angles between vertebrae to kind of get a sense of the gravity of a patient's scoliosis. And the last X-ray product we built is called BoneAge, and it does the job of regressing the apparent age of the bone so that it can be compared to the physiological age of the patient and allow you to detect abnormality in patient growth. Thanks to our 27 million fundraising last year, we're expanding these products to other modalities. First to breast imaging, where we're going to try to detect breast cancer on both mammographies and tomographies, and also on CT images, where we're working on detecting lung nodules and bone metastases. So I want to zoom in a little bit on one of these products, and I want to talk more about BoneView, which is our flagship product, which we have the most material on. And I want to give you a sense of the levels of performance we were able to reach. So I took the graphs, one of our clinical study that we did on BoneView that was published in Radiology. So we have 480 exams, 3 MSK experts that did the gold standard, 24 readers. You have here the RAC curve. You can see the performance of the AI. The cross is the practitioners, the performance of the practitioners without the algorithm. The circle is their performance with it. And we see that we're able to, the first thing we see is the AI alone is already better than the practitioners. And the most interesting thing is that when the practitioners have the tool, they get better performance, even better than the AI alone. I put some key figures. It's really, really, really good to see these. We can really, really diminish the number of missed fractures. We increase sensitivity by a large margin. And we also diminish reading time, which is very, very important for an industry where you have a shortage of radiologists and ever-increasing number of medical images to interpret. I put also some KPIs as to show how we scaled the deployment. We are now in more than 1,200 institutions in 34 countries, which means that we are able to build a product that we can ship worldwide and that generalizes across different populations. And we process more than 20 million exams per year for that product. So MSK X-rays, pretty much. Okay, so how did we get there? As you might imagine, it takes quite a bit of effort. It took us 3 years to reach a stable version for BoneView. So by stable, I mean excellent performance across the board, subpopulations, subgroups, bones, etc. So thousands of AI experiments, thousands and thousands of regulatory documentation, and hundreds of user interviews. I want to focus on the data, which is one of the, the most demanding asset to build. So we labeled over 300,000 images, and that totals over 1 million instance annotations, so bounding boxes or segmentation masks, which are very, very expensive to get. Most of these instances have additional parameters that are also annotated, so the type of fracture, is it displaced or not, etc. And we had to do that on our own in-house platform because at the time, and still today to some extent, if you want to be very precise about how you annotate and you want the workflow to go exactly your way, you need to build your own platform still. And from a clinical standpoint, we had to do a lot to prove and show everyone how well the product behaves. So we did more than 18 clinical studies. So some are MRMC, multiple reader, multiple cases, like the one I showed you before, which are very expensive to make, especially in the US. They cover 5 countries, and we also have 4 targeted studies for very specific subtypes, types of fracture, locations of fracture, like scaphoid fractures, which, if not detected, can lead to very, very complicated situations for the patients. So that's what it took to build BoneView to a satisfying point. And our mission continues. We're only getting started. We need to continue to improve the performance of these products, especially out of distribution, to be able to scale better. As I said, we're extending the Copilot to new modalities, new pathologies, and we really, really need to accelerate the building of these applications because it costs a lot of money, and the need is there, and we can't rely on VC money that much through the whole life of the company. So we need to be really, really efficient on how we build these tools. And for that, we have started investing dedicated amounts of time to research for a little more than a year now. And I want to talk a bit about that. So we've been looking at foundation models. Let me give you a bit of context as to how I see things there. We used to do fine-tuning or transfer from pre-trained models, so typical stuff trained on ImageNet 10 million or ImageNet 10K. Takes 3 days on 8v100 to build. We didn't build them ourselves, we just used them off the shelf. And it's trained with supervised criteria. And there's been a paradigm shift. We've moved from these pre-trained models to the foundation models, or the visual foundation models in our case, which inherit that definition, that name, from the fact that they can be really performant on a very wide variety of tasks with certain very interesting properties. And these were able to emerge because we've gathered sufficient amounts of data in the order of magnitudes higher than before, tens, hundreds, or even billions of images. We have much more efficient compute in higher quantity, so we can train these models 3 days on tens or hundreds of thousands of A100 GPUs, and we have the advent of self-supervised criteria to help us do that and to allow these models to exist. So the first question is, regarding foundation models, it does require a certain amount of data, so do we have the data? Just a word on open source data, which is a very good thing regarding radiology in general. You are able to gather quite some amount of let's say, chest x-rays. You can go up to approximately 1 million chest x-rays just gathering public data. That's really helpful because it laid the groundwork for researchers and for everyone to start working on these foundation models without having to work too hard on acquiring the private and data in such amounts. And what's more interesting is that at Gleamer, we're fortunate enough to have been building these products for 6 years now, so we organically and continuously built a very, very large data asset, which allows us to be in a position today where we have more than 1 million, sometimes much more, images available to us for training for each of these modalities that we're thinking about. So that's very interesting. That puts us in a very good position to work on these foundation models. And I should also say that most of the images we have come with associated radiology reports, which might be interesting depending on what you want to use for training. So what can we hope for when looking at foundation models, especially in the field of radiology, and with the data regimes that we're talking about, so a couple millions of images? I just wanted to highlight a few papers that we based our analysis on before diving into the subject. These 4 papers, so Remedis is one of the first radiology foundation model. It's from Google, it's from late 2022. They do this 2-stage pre-training where they first do a very large-scale pre-training on natural images. This is where you're on the very large data scale, tens or more than tens, so hundreds of millions of images. And then they continue the pre-training in the medical domain with public images for chest, breast, and the 5 other modalities that they do, where they have a lower number of images, but still pretty big, so around 1 million, or that's pretty much the order of magnitude. Then there's Elixir from Google as well, that follows the same 2-stage process, except that they use CLIP, and they're able to do that because we're fortunate enough to have MIMIC, which is a chest X-ray public data set for which you have associated reports, so you can contrast the image in the reports. And then later in 2023, RedFound came out from the NHS, which published in Nature, a very nice paper. They ablate the fact that it's better to do the 2-stage process instead of doing the pre-training on the retinal images from scratch, which is very interesting and kind of guided us in our research. At least they show that that's what works better in their use case. And finally, early 2024, RadDino came out, as well as RayDino more recently. So these models kind of moved to the more recent self-supervised criteria, which kind of established itself as the go-to visual criteria if you want to train your own foundation model, which is Dino v2. And so all of these 4 papers show some very interesting properties. They all kind of hint at them in some different ways. One of the most important is the dramatically improved label efficiency. As I discussed earlier, it takes a lot of time and money to get the data to build a satisfying product. For in the example of BoneView, mentioned 300,000 labeled images. If we're able to reduce that by a significant factor, we're cutting the costs of development of these products. So that's obviously interesting to us, especially for new applications. They also hint at increased out-of-distribution performance, especially if you keep the backbone frozen, and especially if the pre-training distribution is wider than the fine-tuning distribution. So that could be interesting as well, especially when you're trying to build a radiology model that can work across populations and across manufacturers, et cetera, et cetera. You do get equal or higher in distribution performance, so that means that if you have a very well-established benchmark, for example, BoneView, 300,000 labeled images, you probably won't get any performance boost, but you won't degrade performance either. And finally, you also get semantically or spatially rich embeddings, depending on which criteria you used. If you did CLIP, then you can align text and images. You can do semantic search in your database. That can be quite interesting. And if you did Dino v2, for example, you're going to get really spatially organized embeddings, and you can do patch similarity if you want to go look for some specific examples of one of the failure cases of your product. So that's what we're hoping for. That's what we're working on right now. And I'll be glad to share results later this year through whatever means we are able to do. And we also want to get this published, so I don't know, maybe in MIDL next year. And that's it for me. Thanks for listening. And I'll be taking any questions if you have any.
Thank you very much for the presentation and for the support. Do we have any question from the audience? Yes, I can see there.
Hi, thank you for your talk. Very interesting. I'm wondering, these huge foundation models are good for performance, but in terms of inference, of running it in clinic? Do you expect any problems?
So we're looking at, so VIT large is what we kind of want to stop at. And we're getting really good performance with VIT base and VIT large. We're seeing the data efficiency gains already, so it's going to be interesting, and the inference time is not going to be a problem.
All right, but do you expect that is sort of the threshold, and above that, it would not make sense in terms of inference?
You can train the foundation models, larger foundation models, so large and then above, huge and giant. But then you'd probably want to distill them into smaller models for production. And you do get gains by distilling bigger foundation models rather than training them from scratch in a smaller size.
All right, thank you.
I think we had one more here in front.
Thank you for the presentation. I just have a question because you present the product BoneView. I'm wondering, how do you actually build your ground truth? How do you do the labeling? Because for some...
Sorry, I can't hear you that well.
Sorry. I'm wondering, how do you actually build the ground truth? Because I think some of the fracture can be more difficult to see than others.
You mean for the study or in general?
In general.
In general. So for fracture detection, usually an aggregate of radiologist opinion is sufficient. We do have a living body of labels that we evolve through time. We clean, etc., etc. We use the AIs and stuff to improve the labels over time. For lung, it's much more complicated because the pathologies are harder to detect. The agreement between the annotators is much less good. So we do use CT scans as a way to ensure ground truth quality.
And another question, because I know there are other products out there that are similar to your products, like the fracture detection and bone age and so forth. I'm just wondering what sets your product apart from your competitors?
So we've always been ahead performance-wise on the fracture detection problem, and that's what put us in the leading position on that axis. Performance are converging over time, but we are expanding our products and building more so that we can get a comprehensive offer for radiologists and to bundle all that into one co-pilot.
Thank you.
One more, Nabil, behind you.
I have a question. How do you have particular strategies to tackle imbalance of data? Because I guess if you have 100,000 images, only very few of them will have fractures. Are there any strategies?
So all the common tricks, weighting, etc. We also try to be very, very careful when we select data for annotation, and we try to cover and have representative and balanced data sets. Or imbalanced, if that's useful to us. Some specific types of fracture are very rare, and we really want to have them overrepresented in the data set for the model to be really good on them. So it's both tricks at modeling time and being very careful when annotating and selecting data.
Thank you.
Yeah, thanks. I'm curious about the scaling of the company. It's impressive. In 3 years, 1,000 deployments in hospital and institutions. So did you partner with scanner vendors? And also, do you have in-premise infrastructure, or everything is cloud-based?
So we try to do as much cloud as we can. Some institutions are still a bit they don't want to get their data on the cloud. So we do have some on-premise deployment, but we try to minimize that as much as we can. And we do integrate the product into some radiology tables. We have something going on with Fuji, which they do sell our product directly with a small GPU inside the radiology table.
Okay, cool. Thanks.
One more here, yeah.
Thank you. Really nice work. Within just short period of time, you got really lots of product launch. I have a question regarding the data efficiency. You talk about so with pre-trained model, foundation model you can apply on the specific task. It's a much shorter time to market, I guess.
Mhm.
So how efficient we're talking about? I just want to get a rough feeling like.
Really depends on the benchmark, but we're seeing things as, as much as explaining 90% of the performance with 20%,
okay.
Of the label data,
okay.
or even higher.
Yeah, that's really impressive. Yeah, thank you.
Okay, maybe one last question here, and we can move forward.
Hello, thanks for the presentation. So can I ask a question about annotation process? So is that like a fully manual, or could you share some insights how to facilitate or speed up the annotation process?
That's trade secrets you want. We do, yeah, as I said, you should think of your labels as, evolving through time as a living thing, and use whatever means you can to clean them up, improve them over time organically. I won't be sharing any specific tricks, but I'm sure you can imagine many ways of doing that.
Okay, thank you very much once more for your presentation.
Thank you, my pleasure.
And we can continue actually with our second presentation, which is actually Oaking, who is a silver sponsor for this year's MIDL. And the presentation will be actually Melanie Lubrano, who is a senior AI researcher in Oaking, will give the presentation.
Yes, hello everyone. So I'm Melanie Lubrano. I'm a senior data scientist at Oakin. I'm working in the medical imaging team. And I'm going to introduce you a bit to what we do at Oakin and yes, what we do at Oakin. First, a few words about our mission, what we are trying to do. It's to unlock, understand complex biology through AI to find the right treatment for every patient. So how do we do that? We combine AI and complex biomedical data to find new treatments, to de-risk and optimize clinical trials, and to optimize and improve diagnosis. If we look at some key facts, we are around 360 employees. Half of them are actually working in the R&D department, and we are 90 data scientists, which is a lot. We are in 6 different offices. The main office is in Paris here. We are working with top pharmas. We have strategic alliances with Sanofi, BMS, MSD. And we have, over the last year, quite significant number of scientific publications in top scientific journals. If we look a bit more into the publication we do, we see that there are quite few of them that are related to digital pathology, and this is what I'm working on at Oakin. We are indeed key contributors to the field since quite a few years, and we have we use digital pathology data, so histopathology, to develop a different type of model. So to give you maybe a few examples, we have models that are doing diagnostics. So we have one model that is called MS INTRIT that is able to predict what we call microsatellite instability directly from H&E wall slides images. And microsatellite instability, it's a biomarker for treatment response in colorectal cancer. We have another model that is called relapse risk that is able to predict the risk of relapse from patients having breast cancer, also directly from the H&E slides. And we have other models. For instance, we have one that is able to predict directly also from the H&E slides some RNA sequencing profile, which is quite nice. So of course, and you have seen some example before, there are quite some challenges when we deal with digital pathology data. The first one is that these data are very big. They are super large images that we can't feed directly into traditional neural networks. We have to cut them into smaller pieces that we call tiles. But the problem is that we usually don't have annotation for all these individual tiles. We only have one global label. And so we are in what we call weakly supervised setting. And the other challenge that comes with this kind of data is the fact that also because we have a very large number of tiles, we don't usually train end-to-end all our model, and in particular the feature extractors, the CNN parts. They are usually frozen and initialized with pre-trained weights like ImageNet or foundation models, as you heard about. And so today I wanted to make a focus, like my colleague at Gillimer, about foundation models and Hawkin first foundation model for digital pathology. Maybe initiate a discussion around that. So at Hawkin we developed a foundation model that we called FICON. It was developed by some colleagues of mine. I can name them, Alexandre Filliot and Jean-Baptiste Chirati, and all the other co-authors on this list. It was a model that was published in 2023. So I'm mentioning the date because the field goes quite fast. And so since then there were quite other new foundation model for histopathology that came up. But our model is based on vision transformers. And they are trained with a SSL method that is called iBot. You can find all the details in the paper. What is interesting to know is that it was trained on 43 million of histology tiles, which is a lot, and it was addressing 17 cancer, 17 indications. So our model is a pan-cancer model. What is also interesting to note is that the weights are available on Hugging Face. The code is available on GitHub, and you can use them and test them under a non-commercial license if you want. In the paper, you can find a very thorough benchmark that we did against the state-of-the-art at the time. And we benchmark our foundation model for 15 downstream tasks. So by using this FICON model, we try to predict from more slide images histological subtypes, genomic alterations, overall survival, and so on. You have all the details in the paper. But maybe what is nice to note is that thanks to FICON and thanks to Schauder, which is also an architecture that was developed by Hawkin, an architecture able to handle these digital slides, we were ranked first at the Kaggle competition at the beginning of the year, in January. So the goal of the competition was to classify whole slides images between different ovarian cancer subtypes. But the difficulty was that we had this unknown outlier class with a different kind of outlier falling in this category that we had to identify with our model. So we were happy to see that thanks to FICON, we were ranked, FICON and the work of the team, of course, we were ranked first at this competition. And if we go back now a bit to a foundation model more in general, we have seen that for digital pathology, it's very critical and very interesting to have a good feature extraction initialized with good foundation model. And of course, we are not the only ones to think that. This is a really random sampling of the last foundation model that was proposed in the last few months. So not a rigorous literature review, but you see that there are many of them. They all have quite interesting names. They are all more and more robust, more and more powerful, trained on more and more data. sometimes hundreds of thousands world slides images, sometimes even millions of world slides images. So it means billions of histology tiles. And so this model will are really the key to have better performing downstream tasks. So if we take a step back, what are we actually expecting from this foundation model? We want them to be able to encapsulate and to abstract the most generic information, the most relevant information for our downstream tasks from the data they were trained on. So the task we want to use them on is, for instance, in the case of Okin, it will be a prediction of histological subtypes, it will be diagnostics, it will be prediction of molecular alterations, and so on. We really want a model able to capture from the imaging data relevant information for these tasks. And what we believe at Okin is that it actually in biomedical applications, we have actually a good opportunity with all the different kind of modality of data we have access to. So we have histology, but we also have different kind of data. We have genomics, we have proteomics, we have different kind of imaging, we have patient data. And the idea is maybe to think foundation model a bit differently instead of focusing on one modality. See if we can maybe combine these different modalities into one model, and see if the abstraction and the knowledge that is extracted from these different modalities at different scale will lead to a better representations of the data. And so this project, this view, is carried by Bioptimus. Bioptimus, it's a spin-off that was launched by Okin a few months ago, so a young startup. And the goal of Bioptimus team is to build this kind of model, a big holistic model that will encapsulate all these different scale and all these different data modalities into one foundation model. And we can imagine that maybe this model, having learned on all these different kind of data, will be able to connect endpoints that usually we don't necessarily connect. So, for instance, answering questions like why a particular DNA mutation is actually impacting the response to a treatment from a patient, and drawing a direct link in some latent space between these two endpoints. And so this is my last slides to really emphasize that we are into multimodality at Okin. We have launched a few months ago, maybe a year ago, this global initiative that is called Mosaic. And the goal of Mosaic, which is a project that we lead with other partners, other academic partners, is to build the world's largest spatial omics data and multimodal dataset in oncology. So at the end of this project, we will have gathered spatial transcriptomics, imaging data, omics data from 70,000 patients, spanning 7 cancer indications. And we really believe that it's by combining AI and this multimodal data that we'll be able to discover impactful insight for the patient, that will actually have a power to make a difference, to find new treatments for patients. Thank you very much for your attention. Thank you.
Thank you very much, Melanie, for the presentation. So do we have questions from the audience? We have one there, yeah.
Thank you. Thank you very much for the presentation. I have a question on this multi-omics initiative. Is there because you have so many omics, different omics, is the most promising ones that you want to combine first? Because I imagine you wouldn't make a model combining all of them at the same time at the beginning, at least.
So your question is, do we select the omics data we want to look at first before combining everything?
Yes, like maybe it's imaging and transcriptomics data that are most promising together or something like this.
Yeah, so of course so what I presented is a very broad and generic vision and the ultimate goal, but there are of course many steps, and the first step is indeed maybe to look at this very rich data that is a spatial transcriptomics that is combining histology and in a simple way histology and transcriptomics. And having a model able to abstract this kind of data would be already a great thing. I don't know if I answered your question, but
yes, thank you very much.
Hi, thank you very much for the presentation. Very nice. I have a related question about spatial transcriptomic data. So I understand it will be very expensive for every patient to receive the similar kind of single-cell DNA RNA-seq data. What do you deal with people with differences in these data sets? Some might have the transcriptome.
Sorry, I don't hear you well. Can you repeat your question?
So I'm wondering if patients don't have the same type of data for the spatial, how do you deal with that? And also the cost, if you can comment on that. Thank you.
Yeah, so in the Mosaic project, the Mosaic initiative, we will have all the needed modality for all the patients we are targeting. So this will be a complete data set, and this is really the goal of this project is to build this complete data set. And then, of course, it will be super difficult to have some super costly modalities for patients, and that's why we are developing a diagnostic model, for instance, that are only using H&E modality, and we will build other models to deal with the data we have. But it's a separate issue from this Mosaic project. Was it your question?
Cost, maybe, but I guess
the cost of what is the cost of gathering these data sets? I have no idea.
Yeah. Throughout the years, you have developed some nice products. I was wondering if they're being used in clinical practice nowadays, and if not, in your opinion, what's the biggest blocker right now preventing their clinical adoption?
Yeah, so we have one product, MS-INTRI, that is CE marked, so it can be used in Europe to make diagnosis. And we have others coming. So I mentioned relapse risk, and we have others coming. The challenge is to have the right product, answering the right questions, and also complying to all the regulation and having all the right validation to make sure the product is able to compete with the usual diagnosis tool or replace it if, in a proper way if there is no other solutions. So we have one for the moment. We will have other for histopathology.
Thanks.
So one small question. The foundation model that you are trained, the foundation model that you train for pathology is at the patch level, as many of the other foundation models, as UNI and Virchow and all the others. Are you looking at how to model the full slide, like maybe with sequence models or space state models like Mamba or these more long context models, or that's the first question, and the second is.
Sorry, can you repeat the question?
That the foundation model that you train is at patch level. Are you looking at whole slide image level foundation models now?
So the foundation model FICON is trained at tile level, and your question is, are planning to look at representation at slide level?
Correct.
Okay. We have other foundation model that we keep internally for the moment that we are still developing, and for sure it's a nice I think a nice very interesting things to be able to capture this slide representation into a foundation model, and I think that the BioOptimus team is for sure looking at this technique and improvement of the model.
Nice. And the second question is, have you looked at a prototype or interaction of the pathologies with this foundation model through like copilot style, like similar to what we saw before, like maybe with an text interface or some sort of interface that allow them to inquire what the foundation model has learned?
So do we have a sort of ChatGPT linking to our foundation model we don't. There is a nice paper and a demo that came out recently.
Chat, right? Yeah.
Yes, that is doing that, and I think it's very interesting and great. But ours don't, and I don't know if we are going to look into that. But yeah.
Okay, thanks.
Hello. So for the Mosaic initiative, with what medical institutions are you collaborating there to get the data from? And follow-up question, do you plan to open source part of that data set, or is it going to be in-house?
So you can find on the OKIN website all the details of the initiative and all the partners we are working with. I cannot list them. I don't have them in mind, all of them, but there are quite maybe 5 to 10 partners that we are working with to gather all the data and process it. I don't know about open sourcing it. I don't know. I cannot answer.
Thanks.
Okay, one last question, please.
I'd like to ask you, the wrong diagnosis may lead to wrong treatment. Do you take the responsibility for mistakes at your company, and what happens if a mistake comes?
Of course, yeah. The model we build, they are not perfect, but often the question we address is a question that is that doesn't necessarily have an answer yet, and so even a partially accurate solution is better than nothing sometimes. So this is one way to answer, and the other way to answer, for instance, for the diagnosis model we are building. So here we, for instance, MS in Treat, we predict the microsatellite instability from the slides for colorectal patient cancer if there are MSI. So they have these biomarkers, they will benefit better from a particular treatment. So our tool, how it works, is not by predicting if the patient is MSI or not, is to say I'm sure that this patient is not MSI, and to rule out negative cases for which we are sure. And at the end, the hospital will double check the results of the positive ones, the ones that we have qualified as being maybe MSI, by doing the traditional PCR test, for instance, to detect this molecular biomarker. So we are building the tool in order to avoid to have any misclassification, any patient misdiagnosed because of the model.
Okay, thank you very much once
Thank
more.
Thank you very much. Now we'll be here today.
And with that, I think we can close this session. And now we are a little bit late, but we can start with a coffee break. And then the first poster session that they will be. Again, once more, the orals will be held inside the auditorium, and the rest of the posters, you can find them outside. Thank you very much, and see you around.
Hello? Hello? Yes, it works. So hello again, everyone. We're going to start this afternoon session with our first keynote speaker. And so we are greatly honored to welcome Gaël Varocco. So welcome, Gaël. So Gaël Varocco is a researcher director at INRIA. He's the leader of the SODA teams. His work covers fundamentals of AI, statistical learning, NLP with the application to health, and lately public health and epidemiology. He also co-founded scikit-learn, which is one of the reference machine learning toolbox, and he also contributed to other tools in Python. His academic journey has led him through l'École Normale Supérieure, UC Berkeley, McGill, and University of Florence. And I think this is it. So, Gaël, it's up to you now. Thank you.
Thank you very much. Ooh, yes, I can probably handle this computer. Well, probably not. Fantastic, merci beaucoup. All right, thank you very much. Hi, everyone. Thanks a lot for having me today. Thanks a lot for to the organizers. It's always really good to have a meeting where everybody gets around and has discussion. So today I'd like to give you a point of view, a personal point of view on the challenges that I've witnessed that we face, from going from the prediction to actually having some impact in the clinic. And the starting point is that it's often the case that predictors that look good in publications actually do not bring any medical or health benefits. It's been shown, for instance, looking at machine learning for COVID in the publication I'm referencing here, but it's a fairly general thing. Now, of course, we need to take this with a grain of salt because actually having medical benefits is hard. It's really hard. But still, I think it's useful to start and take this as a challenge. And to think, how can we think about this problem, and move forward. And the first thing I'd like to talk about is the fact that there is often a gap between the data and the application. I'd like to dig a bit into this and to show and suggest things that we can do to narrow that gap. And so, a few years ago with Veronica, we challenged ourselves to, think about, where had we failed? Where were the challenges that we, despite, working on this general scope of machine learning and medical imaging, where were the challenges that we hadn't addressed? And looking at surveying problems that were documented, something that comes up and is well documented is that one might have a good prediction, a legitimately good prediction, but it being useless. For instance, because this prediction builds on the consequences of the diagnostic. So to give you a specific example, I'm diagnosing, or people are diagnosing pneumothorax from chest X-ray, and what they're actually using, what the AI ends up using, is a chest strain. Now, once the clinician has put the chest strain in the person, it means the diagnostic of pneumothorax is there. So the chest strain is a consequence of the diagnostic of pneumothorax. So that's not a very useful prediction. It's also true, for instance, people have shown that looking at classifying whether skin lesions were malign or not, AIs would detect that in the training base, the malign skin lesions had been circled out. Now that's very nice, it works really well, but that's not useful to diagnose the non-circled out malign lesion. So that's the first class of problems. And then another actually related class of problems is because of sampling bias. Because basically the data that people are working on is not representative of the target population. Technically, the first problem is a specific sampling bias. But it could be all kinds of things. And I think that sampling bias is very present in a lot of what we do. For instance, we may work on images that are of too high quality because that's what we have, or because that's, you know, what works. And so when we do these things, we're focusing on what we might call internal validity. So our prediction works and our study is internally valid, but it doesn't carry out to a broader world. So there's no external validity. So that focus on internal versus external validity is a typical academic focus that will give us good predictions. But, you know, because we're seeking good predictions, we might actually focus on beautiful data, or on the data that we can have, rather than on the data that's present in the wild. Of course, you know, there's a lot in those statements. You know, what's data in the wild? That completely depends on where you're setting. So those notions are blurry, but they're quite fundamental. And so in general, we can think that we have things that are selection biases. So basically, we have an underlying source of data generating process, and we're only selecting part of those samples. And some selection biases will break the association between the data and the target that we're trying to predict. I'm going to call the target the outcome, because that's the kind of vocabulary that we use in health. But outcome and target, I guess, means the same thing for now. So my outcome is Y, my data is X. And some selection bias, for instance, selecting on a common cause to Y and X, will break the association. So this is a selection bias. My orange cloud of points is actually selected from my blue cloud of points. But you can see that the association is not the same. So the association that I learned will not carry over from the orange to the blue. So it's important to keep in mind that you know, acquiring more data, having bigger models will not solve this problem. This is not a problem of more data, bigger model. It's not a problem of more of what we typically do. And just to not be too abstract, I'll now expand in a couple of common cases of these selection biases. And I'll start with one that is, a very, very classic problem of health. It's one of the seminal statistical problems, which is the problems of censored data. Here, the idea is that the outcomes have not yet been observed. I'll expand, and it is related to something that is well known under the term of survival analysis. And so to detail specifically survival analysis, the broader scope of survival analysis, we're looking at individuals and we're following them. Why are we following individuals? Where if we do health, we should probably not only worry about diagnostic, we should worry about prognostics. So, the future of an individual. So for this, we need to follow the individual. And we can follow the individuals only for a period of time because we have a limited budget to do the study, because some people will move out, they'll move houses and we'll lose them. And so we're looking, for instance, we're studying a specific event, a health event, for instance, the onset of cancer. It typically takes a while for people to develop cancer. And so if people move out, or if our study is too short, we will not detect people who will have cancer. I mean, I could even push this even further. We might be studying death, all-cause death. Most people die at some point. You just have to wait long enough. But if we're, looking too narrow, we're missing this. So that's the general scope of survival analysis. On some data we have observed the event, the outcome, and so on some data we haven't observed the outcome because we haven't waited long enough. So we call this data censored. And there's a specific subset of survival analysis which is known as competing risks. Here we're looking at multiple different types of outcomes. So you can think of it as a multi-class survival problem. Okay? And so typically, in this realm of problems, we might have two questions. We might have what we might call a time to event question. You know, how long does it take for the event to arrive? Or a type of event, type of outcome question, you know, are people more likely to die of a car crash or of a cancer? So that's more the competing risk question. And so if you do naive analysis, it's biased. And I can come back to a story that I lived during COVID, where we were doing data analysis and politicians would come online, you know, 1 week after the beginning of the outbreak and say, you know, it's fine. The illness doesn't last more than 1 week. Now that's quite obvious because, you know, people haven't actually acquired the illness very, very recently. So, you know, you waited another week, so it was 2 weeks after the start of the outbreak, and people would say it's a 2-week-long disease. 3 weeks, it's a 3-week-long disease. And so we were tasked to forecast how long people would stay in the hospitals because this, you know, you had a hospital planning problem here. And so that's typically the setting for survival analysis. And that's typically the setting where if you do it naively and you just take, you know, how long everybody has stayed in the hospital and you average, you get something that's meaningless. Okay? So that's a specific and very marked case of selection bias. I'm not really going to go in the math. I decided not to, but so we recently looked at this with Julie Alberge, one of my students. And so there is quite a literature on this problem. And surprisingly, what we found is that there were still fairly fundamental things to be done. And what we found is that approaches, I don't want to get too much in the detail because I suspect you're not experts of survival analysis, but approaches that are fairly natural, fairly simple, hadn't been very studied. And there was actually a lot of benefit to be had on those approaches. And specifically, the kind of approaches that we studied are fairly natural approaches in this class of selection bias problems where you're computing the probability of censoring. Okay, so you're computing the probability of being censored. That increases with time. At some point, by the way, that probability might be 100% if your study is 3 weeks long. After 3 weeks, everybody is censored, okay? And then you weigh the observed sample by the inverse probability, okay? So it means I'm going to take my standard, so I'm trying to predict, for instance, competing risk, I'm trying to predict the outcome, I'm trying to do standard survival analysis, I'm trying to predict the length of stay, and I will do my standard machine learning to predict from my data, but I will weigh by the inverse probability of censoring. That's a fairly natural thing to do. What we found is that people hadn't really studied this a lot, so we were able to show that this was technically a proper scoring rule, which means that it recovers the true outcome probabilities. But also compared to what people have been doing other things that really make sense. I don't want to go in details, but people have been doing things that are called hazard models that are related to the continuous view of, it's the time derivative of the event. But those hazard models actually create coupled losses that can't be used well in stochastic solvers. Whereas our approach, which is technically more naive, actually decouples the loss, which means it can be used in a stochastic solver. So we didn't apply this in deep learning because we work on electronic health records and gradient boosting is typically what works best here. But technically the loss that we have here can be applied to deep learning, whereas the previous ones require dedicated solvers. And the fun thing is that at the end of the game, at the end of the day, we get something that empirically is both significantly faster than most of the things that have been published because they're using complicated optimization schemes. And on top of this, recovers better the statistical truth. I don't want to get in details of how we evaluate this. It's complicated because we need to compensate for the censoring in the evaluation. But what you can see is that we achieved a really good trade-off where one of the fastest methods and it's the best performing method in terms of integrated bias scores, so statistical performance. Okay, so why am I talking about this? To give you an example of how we can take a machine learning problem and fix it, basically. And that's here what we're doing is we're fixing the loss to account for the censoring in the specific case of survival, but the problem is fairly general. Okay, so now if we take a step back and we think about how our models are used. Our models are typically, the prediction of our models is typically used to support a decision. So we may have, for instance, a model that predicts a health outcome given a health covariate. So here I'm putting only one health covariate, but it could be something much more complicated. And so how can I use this to orient intervention choices? Well, if in the model we're modeling the intervention, where we say, on some people I have had the treatment, and on some people I haven't had the treatment. So then I can compare on an individual treated, the prediction of the model should the individual be treated versus the prediction of the model should the individual not be treated. And then I could use this as a decision support system. This is, by the way, how many clinicians are using prognostic models. They're really, they're taking the model and they're plugging in the model. Well, what if I would decrease the blood pressure by 1 unit? Would that improve the health outcome for the patient? Yes or no? So it's intuitive reasoning. This can be related to a formal causal inference framework, which is known as the potential outcome framework. So the potential outcome framework, the idea is that we have here 2 potential outcomes, and the untreated outcome, why not, and the treated outcome, why one. And we're going to model why not and why one as a function of the covariates X. And then for decision making, the proper quantity is what's known as the individual treatment effect, which is comparing the predicted, treated versus untreated outcome for the same individual. Okay, so this is, I'm summarizing, but there's a whole body of literature around causal inference here. It's nice because in this body of literature, we can reuse our machine learning tools. Now we have one challenge, is that we're not observing both outcomes for individual. I can't observe the treated and non-treated outcome for the individual. So we're observing only one potential outcome, and statistically, what we're doing when we're using machine learning is we're extrapolating across individuals, right? We're saying, I'm going to look at all the treated individuals, and I'm going to be able to extrapolate the effect of the treatment as a function of the covariates, and vice versa for the non-treated. Now we have one problem here, is that the treated individuals and the non-treated individuals may not be comparable. So the healthy individuals may not have received the treatment. And once again, this can be seen as a selection bias. This is a selection bias to a balanced intervention distribution. Here I'm using the word balance, but if I wanted to be statistically rigorous, I should say a random. Because if I have a random attribution of the treatment, then by construction, on average, treated and untreated individuals are comparable. So that's the basis of randomized controlled trial. And so if we want to get to individualized treatment effects, so treatment effect as a function of covariates, then we can use machine learning to basically go beyond the average effect that we're getting in randomized controlled trial. But the challenge is that those 2 populations are different. And so when we apply machine learning, we may get good risk minimizers that actually picks up this difference. So this is a real predictor, a random forest predictor. And we can see that the random forest predictor here is just shrinking the untreated. So for the untreated unhealthy, so you can see that there's basically no untreated unhealthy. All the unhealthy people have been treated, but the predictor here is shrinking the prediction and is not fitting this very well. And the reason is that so this is for one predictor. Here's another predictor that technically is a worse predictor. Okay, so these lines are technically worse predictors. They explain less variance of the data. However, they extrapolate better. And what I can tell you, this is simulated data, so what I can tell you is that Here, the worst predictor gives the best causal inference. So here we have a problem, which is that, or if we do machine learning naively, we're going to pick up the association between the outcome and the treatment. We're going to pick up this association via the population difference between the treated and non-treated. And then we're going to have a predictor, which is not a good predictor to reason on the causal interventions. And the underlying problem here is that the error to minimize is not on the observed distribution, but on distribution that we don't have access to, which would be a distribution where we would see jointly Y0 and Y1. And we have a selection bias between those two, and this selection bias is one that is not at random. So we're basically minimizing the wrong error when we're learning the predictors. So this, by the way, is something that has been well studied in the causal inference literature. My group came in late on this, so for instance, the math behind what I'm presenting today have been done by Nie and Wager in a paper in 2021. So we, with Mathieu Dutrolin, were interested in how to select the best predictors. And the problem is that, once again, you can't do standard cross-validation to select the best predictors. And what we found, I'm going to cut a long story short, is that we can reuse tools that are well known from the causal inference literature, is that we can use risks, so measures of errors that use the probability of treatment to compensate for the fact that there's a systematic association between treatment and outcome. And the one that works best, that's detailed in our study. Our study is mostly an empirical study with a few theoretical results. But anyhow, the one that works best is known as the R risk. It has a complicated formula that is basically a squared error of the difference between the predicted outcome and the observed outcome, but it accounts for, it actually rewrites this difference as a function of two things: of the conditional mean outcome. So the conditional mean outcome is the mean across Y1 and Y0, okay, conditionally on X, and the propensity score. The propensity score is the same object that I had previously. It's the probability of being treated. So it's the probability of censoring bias, okay? And mathematically, this stems from the fact that you can rewrite YA, where A is treatment, so A is Y1 or Y0, in a specific decomposition that separates the common cause and the delta between the two. So M is the common cause, it's the conditional mean outcome, and then the other term is the delta between the two. And when you do this, you're able to balance errors. It's not obvious, but you're able to balance errors, and then you're able to rewrite things in a way that errors balance better. So maybe this is taking you quite far in the direction of causal inference. But I just wanted to give this as an example that people don't always use our predictor, the way we think. They use a predictor for a specific goal. Here it's decision making, and this decision making actually is related to causal questions, and the corresponding risk, the corresponding measure of error, is actually quite sophisticated. Well, I think there's a lot to be done here. Now, one more thing in terms of causal inference, I'm going to go back to a problem that I mentioned before. If we use post-treatment, post-intervention information, we have inapplicable prediction. For instance, a drug may lower the blood pressure. If we use data where we've picked up blood pressure from after the drug was given to the individuals, we're actually indirectly picking up the effect of the drug. And this happens more often than you would think, okay? It's just like the chest drain in the pneumothorax. So anytime we're picking up consequences of the intervention, we're building a predictor which cannot be used to reason on the effect of this intervention. Once again, this happens more often than we think. So there's a whole, causal inference framework popularized by Judy Pearl, which sometimes is complicated, but really the most important thing to keep in mind is that when you're going to put variables in your model, you need to think about their causal relation to the question that you're asking. Anything that's a confounder, so a confounder is something that modulates the both the outcomes and the probability of being treated. Anything that's an effect modifier, so it modulates the outcome, you need to put in. Anything that's a consequence of the treatment, such as a collider, an instrumental variable, a mediator, if you put in, you will build an invalid causal model that you cannot use to reason on causal effects. And if you add to this temporal data, such as electronic health records, there are so many subtle effects where you have subtle cutoff effects in terms of how you define your inclusion criteria that actually create selection biases that basically creep in as colliders. And I don't want to go in the details, but with Mathieu Dutroline, we wrote what we hope is a tutorial article on this. And I am going to give you the reception of this tutorial article. The people doing epidemiology basically tell us, duh, everybody knows this, which, by the way, is not true because you can still find those errors often. But still, it's well taught. And the people doing machine learning basically tell us, that's not the question we're asking. I personally think it's a very important question. Anyhow. Okay, so I think if we want to go one step further, and if we want to have not only worry about diagnosis, which is what we typically worry about when we do medical imaging, but if we want to go further and we want to worry about health outcomes and we want to worry about how does the clinician use our predictors for health outcome, it's useful to adopt the causal inference framework. And hopefully, the two papers that I've presented that we try to make didactic are useful as an introduction to this. And there's very little new math in those two papers, or new results. They're new empirical results, but I hope they're useful. Okay, so to kind of summarize a bit this part, the problem we're facing more often than we think is that our data doesn't reflect our application. And here it's not a question of predicting best Y, whatever that Y means given X, which is typically what we worry about when we do machine learning. I've given you the example of survival, where here the problem is that I haven't observed individuals long enough. Causality, I've observed only one potential outcome per individual. And once again, it's not a question of just doing more machine learning, bigger, better. Or if we do this, we need to specifically model those effects. So when I work in health, to be honest, when I work in health, those problems are the number one problems that make me lose sleep, that make me think that what I do may not be useful for health. Okay, so to come back to our problems, predictors often fail to bring medical benefit. There's another aspect to it. It's that our evaluations sometimes are not stringent enough. So I'm going to go a bit fast on this part because I have too many slides and because some of you have already seen it. The first, if we want to do more stringent evaluation, the first question we should ask ourselves is, what are the metrics we should be using? In a sense, I already mentioned for causality, we need to be careful about our metrics. But even for like a simple problem, so I guess, in medical imaging, we typically work on case-control data sets where we have, we're trying to diagnose control versus a specific pathology, and we have 50% control, 50% specific pathology. That's not representative at all of real life, The prevalence or incidence of pathology in real life is very low, and that's a good thing, right? So real life is about real life health is typically about very, very imbalanced setting. So what are the meaningful metrics in such a setting? Well, first, accuracy is not a very meaningful metric, because if I have 90% of class 0, then I can just have a predictor that predicts class 0, and I still have a 90% accuracy. So people use things like balanced accuracy, which is a mixture of specificity and sensitivity. I can't really give a specific meaning to balanced accuracy, to be honest. Rather, what I can do is I can take a step back and I can say, okay, what does sensitivity mean? What does specificity mean? And if we take a step back, specificity measures the probability of the prediction being positive, given the ground truth being positive. Specificity measures the probability of the prediction being negative, given the ground truth being negative. Okay? I'll claim we're asking the wrong question. We're interested in the probability of the truth being positive, given the prediction being positive. This is what we're interested in. And this is not what specificity and sensitivity measure. So we can compute this. We can use Bayes' theorem. And if we do this, we're going to have the prevalence that comes in. And so we can predict we can write the probability of the truth given the prediction. This is called the positive predictive value. That's for the probability of the truth being positive given the prediction being positive, and then you have the negative predictive value. If you look at medical publications, they typically use the PPV and the MPV. I must confess it took me 10 years to understand why. And the reason is because it's asking the other question. None of this is new again. I'm telling you things that have been known for, decades. Now, one reason why people don't like this to benchmark machine learning algorithm is it depends on the prevalence. So it characterizes not only the classifier, but the data set. So one thing that we can do is that we can look at the odds ratio, and I'm not going to go through the math, but there is a specific odds ratio. So the odds of an event, the odds of A, is the probability of A divided by 1 minus the probability of A. So the probability of A divided by the probability of not A. And the nice thing is that there's, those ratios tend to cancel out prevalences. And there's a specific ratio, which is known as the likelihood ratio of the positive class, which is the odds of a test being positive, the odd of a true positive given a predictive positive divided by the odd of a true positive. This thing is actually independent of the prevalence. Why is this important? Because I can compute these things on the case control a cohort, the kind of cohort we typically work with in medical imaging, and then I can extrapolate from this the behavior of the predictor on a variant balanced data set, because the variant balanced data set is going to change the odds of the true positive. Okay, and so then I can compute the odds of true positive given predicted positive on my target population. Okay? So I can extrapolate across test sets of different prevalence. This is what I'm trying to say. Anyhow, if you don't, I'm not expecting you to remember those formulas. They're not very complicated. What I really want to, go in and stress is that you need to think about the fact that what matters is the increase of information that you're bringing to the user. And for this, you need to think in terms of the probability of the truth given the prediction, and that depends on the prevalence. You can always come back to this book chapter that we wrote with Olivier Colliot, who is in the room, and we try to make it didactic. Okay, on to another problem. Working with practitioners, one finds that there is always a demand for not only predicting, whether, an outcome 1 or outcome 0, but also predicting some form of confidence or probability of the outcome. That's very strong demand. And one of the reasons is that every decision in real life is a trade-off. You need to balance harm-benefit, and this trade-off is going to be a situation-specific trade-off. You're going to account for the specificities of the health system on the specific day of the specific individual of the individual's family. And here in machine learning, we talk about calibration. And I want to stress that the definition of calibration is that it controls the average error rate. So a classifier is calibrated if the average error rate for all samples with score S is S. Most people don't have the definition in mind, but that's the definition. That's what the expected calibration error measures. It controls an average error. It does not control an individual error. I can have a classifier that is completely calibrated, that has zero calibration error, that has abysmal control on individual errors because it has substructures, because it has subgroups on which it's overconfident and underconfident. It may be massively overconfident on the males, massively underconfident on the females, and be calibrated. Males and females might be easy to guess. It might be something harder. So it's important to stress that once you have zero calibration error, it's not the end of the story. You do not have good individual, you may not have good individual probabilities. So there is another kind of metrics that control completely the probability of y given x, which is what we're interested in, and those are called proper scoring rule. The challenge of a proper scoring rule is that you're never observing the probabilities. You're only observing discrete events, 0 and 1. And you want to control the probabilities. So the magic of the proper scoring rule is that on expectancy, when the proper scoring rule is minimum, you know that you actually found the underlying conditional probability distribution. Okay? And the proper scoring rules can be decomposed, and one decomposition is decomposing the proper scoring rule in, so you have a predictor, and you're decomposing its error that you're measuring as a proper scoring rule. Oh, I forgot to mention the Breyer score is a proper scoring rule, and it's just a squared error between your discrete labels and your predicted confidence. So it's a very trivial proper scoring rule. It's just a squared error. And you can decompose it in three terms. One is the irreducible error. It's related to the fundamental randomness. It's the aleatoric error. It's, you just can't, this cannot go to 0. And then the remainder is composed of the calibration error and a reminder term, which is known as the grouping error. And what the grouping error does is it actually controls what is not explained by the average in your calibration plot. So the fact that you have individual probabilities that vary and you may have error on the individual probabilities. I just want to stress that in practice, we cannot plot such a calibration plot because we never observe the individual probabilities. This is why it's very hard to estimate the grouping error, because it requires the access to true probability. So to cut a long story short, with Alexandre Pérez-Lebel, we did an estimator of the grouping error, technically a lower band, but in practice it's often tight. And the way we do this is that we compute well-chosen bins, and by computing the well-chosen bins, we can approximate the true probabilities. So I'm going to speed up, but what I want to say here is that when you're thinking probability of outcome, there's too much focus on calibration, and we also need to estimate the remainder, for instance, the grouping loss or a proper scoring rule. I think this is really important. I see a lot of mistakes in machine learning paper that conclude that because they have a small calibration error, they have access to the individual probabilities. It may not be written this way, but the reasoning that people do is that, and this is an incorrect reasoning. We need to go one step further. We have code that computes the grouping loss. It's called GLESS, for grouping loss estimator. Okay, I'm going to go fast on this. I'm running out of time. But one thing that we need to talk about is evaluation noise. Estimating the generalization error is really hard. And we might think, well, we have enough data, it's negligible. But we find what we found with Veronica looking at things like Kaggle competitions is that there is a lot of noise in the evaluation process. And if you take two different evaluation data sets, the order of the algorithms is going to massively, massively change. This is an intrinsic problem. This is related to the fact that we're benchmarking on algorithms on finite amount of data. And so we have sampling error on finite amount of data. You know, if I measure, I can think of my algorithm as a dice that I roll. You know, it may give me the right result. It may give me the wrong result. If I need to evaluate how biased is my dice, if I'm rolling it only 100 times, I don't have a good evaluation of the dice. This creeps in with the incentives. This is a challenge that we ran a few years ago. The incentives were aligned. People were given money if they were to generalize on the hidden test set, but they overfit. So I'm not gonna, I don't have time to go into details, but the noise, the evaluation noise is really important, and it's really important to measure it. And here I tell you the confidence intervals that you can compute with a binomial, and you can see that they're pretty big. And then I can tell you many things about how to measure the confidence error, but I'm sorry I'm lacking time. So I will not cover this. Okay. Now, what we see here is that often we over-evaluate the value of what we do. And that's not new. I mean, it happens everywhere. Industries have always done this. And there's almost a selection bias here. You know, the people who are most confident that what they do is great move up the ladder, and the others stay below. So there's nothing new, but it's a problem. And it couples in with a narrative of the industry, which is that basically, you know, the new things are always solving it, and, you know, we're just, you know, building bigger stuff, and we can see that the bigger stuff that we're building, you know, convolutional neural nets, you know, large language models, are basically always improving on the benchmarks, and we're just going to solve all the problems like this. And, you know, this narrative and this behavior has been going on for decades, and as a consequence, we just move to bigger and bigger and bigger models. We've seen this, but, you know, it's interesting to put numbers on this. And what we see is that, you know, the compute is actually overtaking the largest supercomputers. So we have compute needs, we machine learners have compute needs that go much faster than the hardware, much, much faster. As a consequence, the training costs are going much, much faster. Now, it's less the case in vision than in language, but, you know, we're looking at training costs. Here it's written 1 million. When I talk to industry people, it's more 100 million. So we're looking at training costs in the 100 millions. This, okay, we might say, you know, it's not important because it's done once and for all, but this couples in the inference costs. So running these things is becoming more and more expensive. And that's really threatening the viability of typical usage. Typical usage can no longer be done on premises. It can no longer be done in hospitals. You know, you need to move the data out, which leads to, you know, privacy problems and everything. So I want to challenge what we're doing here, you know, as a community. We're playing a game of scale, and that's the wrong game. We need to choose what we're working on. Bigger is not always better. Beating the benchmark is not always the right solution. When we're doing this, we're going in the value system of the big players. We're going in the value system of the hardware providers, of the cloud providers. We're not working on health problems. We're working on big tech problems. This benefits certain actors. More data benefits social networks. Large compute, once again, benefit hardware makers and providers. And sorry, this may be hard for French people, but it couples in all kinds of things. You know, we want higher quality scanners. Those are only possible in large hospitals. So in big cities, we want, you know, our AI to work on the latest phones, and then they can only be used by rich and urban and young people. So we're, you know, this technology is creating a widening gap between, you know, the rich and the poor, the urban centers and the forgotten rurals. And this is a map of the election results. And if you squint at it, you can see that basically the cities didn't vote far right and all the rest voted far right. And we need to think about these problems, okay? And, you know, going back to the problem I was asking, you know, yeah, it is provocative, but I really do believe it's important. Let's worry about clinical impact. And if we worry about clinical impact, let's focus on what things are useful for rather than how powerful things are. It may not be important to gain 1% on the benchmark. It may be more important to think about how it's being used and the biases of our benchmarks. Thank you.
Thank you very much, Gaël. Any questions? If not, I can start with maybe an easy one. What did you apply your method on, like real data, on health data, for example? Do you have some application examples of your work?
Not much, to be honest. No, I mean, yeah. So we have application examples, but they're not published. These days, I work on electronic health records with the hospitals. We did a lot of work during COVID. It was really interesting. To be honest, the successes that we met were more hospital management successes. You know, being able to do bed allocation. That's the easiest because the biases are less important here. We're working, so it's a line of research I've started, 3-4 years ago, and so we've applied this on health data. The benchmark I was showing you was health data, but this is not a health success; it's a benchmark. My experience is that health successes take much longer. We're spending a significant amount of time working. For instance, one of the problems we're working a lot on is diabetes, where prognosis of diabetes is a complication. We would like it to shape how the kind of actions that we're taking, the follow-up actions we're taking on the people who have diabetes. I can't claim we've had a success. We haven't really published a paper. We've been working for 3 years on this. It's hard.
Yes, a question over there.
Hello, thank you for your presentation. Would you agree that, based on what you said, do you think that we're doing way too oversimplify the post-processing and the ensembling? And could we, based on the metrics that you presented, would it be a region to look into? Instead of looking into a bigger model, we try a smarter way of ensembling models.
I think what goes behind your question is, can we change the metrics we're looking at? And I think that's exactly it. We need to change the metrics we're looking at. And here the challenge and the reason it happens is it's not just a formal problem. The way we change the metric is that we talk to people who have a very different cultures. For instance, we talk to people who do diabetes, and we don't talk the same language, and it takes a lot of time to understand what's a good metric. But from a mathematical perspective, it's about finding the right metric. If we found the right metric, we can optimize it, or we can optimize the expectancy of the error on this metric. From the more like a sociological aspect of sciences, it's bridging the gap between people who speak different language to be able to define those goals together, including the patient associations. And that's the real challenge, I think.
So I had a question regarding the things you showed on the Kaggle datasets challenges, where people overfit on the validation data, and this is actually quite interesting, I think. Could you think about, is there a problem that the validation set is just too small? If our test set is infinitely large, then this problem doesn't happen, and people have to overfit on all the data, and it doesn't work. Is that another conclusion, or is there something else to it?
No, thank you for your question. That's exactly it. The validation set is too small. Now, is it too small or not too small? Do these differences between algorithms make any difference to the application? I don't know, but it's too small to make the difference between the models. And here, we have this tension. We want the model that predicts best, but so we try to save as much data as possible for the training and use little data for the evaluation. But then we're evaluating our models wrong. But yes, so of course, with infinite evaluation data, then this stops being a problem. And that's one reason why I advocate, I didn't have time to cover this, for cross-validation to evaluate models. We don't want to do this because it's costly, but it doesn't make sense not to evaluate our models right. These days we do fixed splitting between train and test. And if we want to evaluate the computer science contribution, we should do cross-validation because it uses our data better to evaluate.
Thanks.
Thanks for your talk. I agree with many points, with benchmarking especially, but I find myself disagreeing about your ending, about large models, because, and my question to you in essence is, would you disagree that the progress that was achieved thanks to scaling up, to making the data set larger and the models larger, that it's a real progress that led to real improvements, not 0.1 dice on some benchmark, but real tangible improvements in the tasks that we're solving. And therefore, it's not like a devil we should defeat, but something that we need to keep in mind in terms of benchmarking and what we really want to achieve in the clinic.
I don't think it's a devil we need to defeat, but I'm questioning some of the progress. We're moving the goalpost. If we go back 10 years ago, we were claiming, 15 years ago, we were claiming that automatic driving would be solved in 5 years. It's 15 years later, and I work with the industry experts on automatic driving, and they think it's going to be solved by economically viable, not I've shown you something that works, in another 10 years. So we're moving the goalposts. Actually what happens is that we've not solved the problems. We've maximized the benchmark performance, but solving the problem requires more than maximizing the performance. It requires things like adversarial robustness to dataset shifts, these things. And we've just moved to, problems that were new each time. We've moved, the AI field has moved mostly from computer vision to large language models. It hasn't solved computer vision. Self-driving cars don't exist. Sorry. So yes, there is progress due to scale. That's true. But what I'm missing is what hasn't been covered by scale. So maybe we're in part agreement.
Yes, I would say, first you need to solve the easiest problem. Let's say that's what's solved by scale, right? If you can't recognize anything in the image, you can't do anything. You can't do fairness.
Why first do we need to solve this? We've not solved the problems we were working on 10 years, so we're just moved on to new problems, but we haven't solved them.
Okay, I think it's a matter of discussion. And for example, the self-driving is solved to some number of nines, 99.99. It's then a question of what is acceptable in the real world. Yeah, so it's an interesting point to discuss, but I think I don't have a question.
Hi, just a more mundane question here. So have you tried any of your survival prediction on data that is more long-term and probably single intervention, things like cancer, for example?
Thanks, Todd, for the question. It's actually the most, the best thing we've tried on is cancer, indeed, long-term cancer. We've tried on a very beautiful data set that's called the CER data set, CEER. It's a long-term follow-up of cancer. For instance, what we haven't done is tried it on medical imaging. But yes, our best evaluation, the paper has 3 evaluations, but our best evaluation, the one I'm most proud of, is on the long-term multi-outcome cancer. Now, it's only a benchmark. Now, the question is whether it can be used for actual prevention. I think there's a lot of work to be added on top of what we did.
Okay. Hello. Oh, Jesus. It's way louder than I expected. I also want to sort of respond on the question we had before last, where I do agree that basically scale will not solve much more anymore, in the sense of bigger models will not perform much better. We're getting really good results on the classic CNNs, for example. I don't really think that vision transformers will add much more. But in the sense of we need to move all these things that we do, we need to move them to clinical practice, and then most of the time for clinical practice you need to certify models, you need to certify pipelines, And then still, unfortunately, often CE certification still requires the DICE score or anything that fits in the metrics, because that's what they'll be looking at. FDA approval will ask for it for the DICE scores, for household distances. So it's sort of built into almost the regulation process that we do need to keep these things going, unfortunately. But the downside of this also, of course, is that I work in the field of segmentation mainly. Segmentation is actually way more subjective than we would want to have. For a tumor segmentation, that's not very well defined. Where does the border lie? So if you have a model that gets a DICE score of 0.95, 0.96, which is great, you get a DICE score on that one particular person's segmentation and maybe not the truth. So how can we sort of fix this, I guess? Because it's built into the regulation process. It's built into trust into the people, basically. Superhuman scores are still based on human labels. So how does...
So there are many aspects to your question. Thank you for your question. So first, kind of like maybe the hopeful thing I can tell you is that there are ways forward, in my opinion. That's my personal opinion, but I really believe in this. I don't really care about a segmentation score. It's a useful proxy. What I really care about is, can you do a randomized control trial that shows me that your segmentation tool, improves a health outcome compared to another segmentation tool. Now, your questions about the regulatory problem is a really, really important one. I'm flying to Shanghai tonight to discuss regulation of AI. You have no idea how important these things are. And it's a very difficult discussion because the stakes are very high and people by now have gotten in a situation where there's a lot of money and people are worried. And so, I put up this slide because what's going on, I didn't really mention this, but what's going on behind this research is a research that tries to ask, how do we build the right evaluation framework for AIs, which can help unlike the regulatory framework. But you used a very good term. You used the term trust. And I want to say that trust is not math. Trust is social discussions. Trust is something social. And we need to play this game. We need to talk to broader people.
Thanks. Hi, thank you so much for the great talk. So you talked about evaluation bias, and you said, okay, one thing we should definitely do is cross-validation and also evaluation on external data sets. Now, if you want to do that in the context of a challenge, this implies that the model needs to be trained in the hardware of the challenge organizer, which again benefits the hardware providers. So how can we ensure more of a fair evaluation in these challenges without requiring the organizer of the challenge to have access to so much hardware?
Okay, very good question, thank you. First, I think that for challenge, and as a challenge organizer, I think that for challenges, we need a left out data. Because if we don't have a left out data, people will probably overfit it if we give them back a signal. Then, in terms of hardware, it's a difficult question. It's a question, it's all in the nuance, right? We can't deny that we need hardware, we need compute. The question is, where do we put the cursor? How do we transform? And it's a question I don't have an answer to, but I'm really interested in how do we transform an improvement in prediction into, in French we call this a medico-economic improvement, or the World Health Organization uses the term reducing the burden of a given pathology. That's a very important question, but at some point, I think we need to question to question whether the metric that we're using actually transforms into an improvement. At some point, we get into diminishing returns. So, the improvement in the metric doesn't actually help. And another thing that I would really like to see, we tried to publish a paper at ICML on this that didn't go through, but is to see the trade-offs. One plot that I really like is a plot where you put, statistical performance. Well, it's the plot I was showing. You plot statistical performance as a function of computational resource. Now, computational resources may mean different things. The size of your model is actually really important because then it doesn't fit on certain devices. But anyhow, you look at this trade-off, and I personally am more interested in something that, improves both aspects than, makes computation worse but performance better. I think we need to incentivize this. So Pareto optimality.
Yeah, thanks again for the talk and the mention. So maybe a bit related to this previous question. So what do you think about open data sets? I mean, from the one side, there's the philosophy of open science, etc., and sharing. But would you say it's also kind of encouraging this overfitting to the benchmarks and not looking beyond that?
Oh, so I'm super convinced that open science and open data sets are super useful. If you don't have them, basically you ground to a halt. What I encourage is skepticism and on all those results, skepticism from everyone, from the reviewer, from the funding agency and everything, and I encourage also, and that's hard, especially on the young people, but I encourage also people to, move outside of this question and ask question, what's the relevant metric and everything. And it's going to be hard because then the reviewer is going to look at your paper and go like, not interesting, because the reviewer is like me, the reviewer is lazy and the reviewer wants bold-faced numbers because it's easier. I mean, yeah, but science is hard. I'm sorry.
Thanks a lot.
Hey, that was a great talk. So, I mean, my question is to you and to the general audience. I think like most of the points that you brought up were relevant to like real world data sets, which actually belong within the hospital setup. And I mean, most of the open source data sets that we play with, I mean, aren't actually having these sorts of problems. So the larger question is like, how should we actually, as a community, how should we start thinking? Because like the hospitals have their own stakes in these private data sets. They don't want to share it out in the public. And I mean, most of these challenges of like continuous learning or, I mean, diversification or robustness to external data sets actually come in the case of private data sets owned by different hospitals. Or if you're moving an algorithm from one hospital to another hospital, and you tend to see these problems, or like diversification in terms of demographics. And so how should we convince, I mean, the computer vision or the machine learning community to stop thinking of open source data sets, or basically how should we bridge this gap between people sitting in the hospitals and the community, the machine learning community actually working on these open source data sets which have less relevance in terms of real world deployment of these, at least so in the medical imaging community?
I'll give two answers here. The first one is none of the empirical results that I showed you were done on hospital data. They were all done on public data set. It's just that we kind of created biases, we did things like this. So we can still ask many very interesting questions on public data set. And the reason why we're doing it on public data set is basically because it's faster, because it allows us to do our science there. Now, another aspect to my question is, let's walk in the hospitals. I'm not kidding. Let's walk in the hospital. It's a long process. It's a trust-building process. The first time I walked into a biostatistician's office and I told him I was doing machine learning, he was almost like, get out. No, seriously. But, you build trust. And that's also, you can see what I'm saying. I'm saying, basically, all the research I've presented here was done on non-hospital data, but let's do hospital data. There's a question, what are the successes we achieved? None. But let's not lie. Let's say we have a fast track that is more of a mathematical and toy problem track where we understand problems, including by creating them in data sets, by simulating them. And then there's a slow track where we talk to the hospitals, we try to work with them. And honestly, the slow track takes forever, but if you look at the dynamics of medical research, it's a much, much slower dynamics than computer science research. This is why we need the two tracks. We're not putting a young person's career in danger by going on a slow track, because that young person also has the fast track. And that young person will be an author on medical papers, but will also be an author on computer science or mathematical papers. That's my view.
Thank you, Gaël. So I think we're going to stop here for the question. Maybe one last time, can you predict our survival rate after this Sunday's election?
Depressing.
Thank you, Gaël. Thank you very much.
Are the speakers of the next session here, and could you come up to the front, please?
All right. After this nice keynote presentation, we have a short but very nice session on representation learning on multimodal methods. So I would like to start with the first presentation, which will be given by Alexander Zolotarev on predicting atrial fibrillation treatment outcome with Siamese multimodal fusion and cardiac digital twins.
Okay. Thank you for the introduction. So hello everyone. My name is Alexander Zolotarev. I'm a postdoctoral research assistant from the Queen Mary University of London. And it's a pleasure for me to be here and to present our research about the cardiac digital twin and their combination with deep learning to predict atrial fibrillation treatment outcome. Let's firstly state the problem. Atrial fibrillation is the most common heart rhythm disorder. And in contrast with usual heart rhythm, which is periodic and originates from the sinus rhythm, from the sinus node, only one source, which leads to the periodic contractions of a heart. As you can see on the left image, on the right image, you can see an image for atrial fibrillation, which has multiple wavelets, multiple pathologic sources of signal. And this is not a life-damaging event, life-damaging disease, but it leads to diminished quality of life and is associated with the high risk of stroke. Unfortunately, the long-term success rate remains low, even after conventional methods of treatment. And this is a problem we try to tackle. So what do I mean by conventional treatment? Usually, the most common treatment is catheter ablation. This term has two words. First one is catheter, which is literally the thin wire, which is located or are located, if there are multiple, inside the artery, the upper part of heart. And then ablation is the same term as in data science. So to ablate means that you need to physically destroy some pathological sources. And if you are not sure where they're located, you can easily do some isolation, so not to destroy the precise location, but to isolate the tissue which is prone to be pathological, and to prevent electrical signals to propagate from this region. And the most common example of it is pulmonary vein isolation, because it is well known that there are some triggers around the pulmonary veins, and if you isolate the ostia of these veins, you can treat this disease like half of all cases. And there are some other techniques, some other strategies for ablation, but unfortunately, all of them are not personalized. So we did exactly the same for all patients, and most of them are based on some anatomical structures. And this is maybe the reason why this long-term success rate is so low. And our idea, the idea of our lab, we try to tackle this problem by creating the personalized cardiac digital twin, and then simulate the electrical activity on it. And you can see these rotational patterns the same as during like clinical cases of atrial fibrillation. And then we try to explore different strategies of ablation on this heart and select the best one. And how it can look like, or what do we need to construct these biophysical simulations for atrial fibrillation. Initially, we need some imaging data from CT or MRI scans. Then we segment left and right atrial cavities, and then create the 3D biatrial surface mesh. Another important step is later we selected specific landmarks on this mesh to do some personalized thing. For example, we can separate the heart into different regions with different conduction parameters, or we can add some personalized structures from other modalities. For example, from DT MRI, we can add fiber orientation. Fibers is like the path for the primary conduction of the signal, because the conduction is anisotropic in the heart. Or another thing I want to focus like on this one, we can add fibrosis from LG MRI data. And this fibrosis it's well known that it plays an important role in atrial fibrillation maintenance. And also it can serve as an anchor for these pathological sources. So if we isolate the regions with the highest fibrosis intensity, we can like do some personalized treatment. This is another type of strategy. And after adding all this information into the model, we finally run finite element modeling and post-process it. For example, here you can see an example of phase singularity density map. Phase singularities define for us the allocation of the rotational activity. And also you can see that this is an image. So by adding the anatomical landmarks, allows us to calculate the universal atrial coordinates to transfer the 3D mesh of arbitrary shape, arbitrary number of units to the 2D images, which is important for data science. But what is wrong? What is the big disadvantage of this? Testing various treatment strategies via these simulations is time-consuming and impractical on the clinical time scales. And ideally, we want to have it fast and useful for the clinicians. And my idea is how can we overcome this limitation is to use deep learning. So imagine we have a patient or virtual patient, and then extract some features from this simulation. For example, phase singularity density map or fibrosis, so they can be either anatomical or physiological, and then run a model to predict something. For example, to predict the probability of atrial fibrillation termination after specific type of ablation. And if we run this model on many cases, on many patients, so-called populational study, we can predict the same even for new patients after running just one personalized model, after running just one atrial fibrillation simulation. So the objective of my study is to develop a novel deep learning pipeline for predicting atrial fibrillation outcome using a large in silico study of 1,000 virtual patients. And this is a pipeline, so we run 1,000 models and then extract from each of them several feature maps: fibrosis, phase singularity density, domain frequency. This is an example of phase analysis. And then we transfer them from 3D meshes to 2D images. And then this is our first bunch of feature sets. Another important thing is that we need to have some information about ablation strategies. So here you can see an example of 4 of them. PVI, you're already familiar with it, and you can see on the mesh 4 rings, or 4 non-conductive rings. We simulate the effect of clinical PVI in this way, so to prevent the electrical signal to propagate from this pulmonary vein ostia. And then we run 3 more ablation strategies, which are left atrial fibrosis ablation, and you can see a lot of ablation lesions, white ablation lesions on the left part of a mesh, then right atrial ablations, fibrosis ablations, and then biatrial ablation. And then for each of them, we have a mask firstly in 3D and then in 2D. This is our first part of feature maps. And then we fed it into a deep learning model to predict the probability. And the good point is that, like good for me, of course, is that we can do it in supervised manner because we can use this mask not only for the deep learning, but also for finite element modeling. So we can run another bunch of simulations to have the ground true, whatever atrial fibrillation terminates or not after a specific type of ablation. So what about the proposed model? After each atrial fibrillation simulation, we had 4 feature maps, 4 for left atria, 4 for right atria. Then we separate them and use the so-called Siamese architecture, 2 heads of them, each contains of dense net, and then we fused the lattice representations of this dense net by a so-called MOB, multimodal arithmetic block, which is really multimodal fusion, which calculate the 4 basic arithmetic operations behind these 2 columns of values: addition, subtraction, product, and division. And then we add some more layers, 2D convolution, fully connected layers, and finally we have a probability. So about the results, first about the data set. After running the finite element modeling for all these cases, for all strategies, we found that the percentage of successful ablation is 40%. And the simplest strategy is PVI, and the effectiveness of it is the lowest, around 25%. And if we ablate more and more regions, the efficacy is 60%. And you can see here on the bottom the example of 5 finite element modeling simulations. Firstly, before any ablation, and then 4 strategies. And as you can see, so initially, for almost all of them, there are some electrical activity, and even after the ablation lesions. But for the last one, and ground true only for this one, is 1, which means atrial fibrillation terminates. And we don't have any electrical activity after 5 seconds of the after initiation of atrial fibrillation. And about the results, so our proposed model, deep learning model, achieved high performance, and our RC is around 0.9. And we try to think maybe we don't need, what about the ablation study, ablation in data science terms? So do we really need all these feature maps? Maybe we can cut some of them and have almost the same results. And it looks like that it's very important to have both anatomical and physiological feature maps. So for example, if you use just only mask, which is only basic information about ablation strategy, our RC is very low. And if you add more and more information, for example, fibrosis alone, or domain frequency alone, or facing density, the metrics are higher and higher, and finally we have the highest performance after adding all of them. And another thing is, do we really need another ablation study? Do we really need this architecture? Maybe we can just simply concatenate everything and have just one head, or maybe we can have this Siamese architecture and to have simple concatenation and didn't fuse the feature maps. And it's also look like that it's not very big improvements, but it's relevant, and it's shown here. So to conclude, atrial fibrillation treatment is a challenging task, and we try to tackle this problem by utilizing cardiac digital twin, and we provide a methodology for calculating multimodal feature maps from these cardiac digital twins, and we also provide a deep learning pipeline that fuses these feature maps. And finally, we have shown that only a full combination of all multimodal feature maps, both mask, anatomical, and physiological features, not both, 3 of them, and also Siamese architecture and fusion block are important to achieve the predicted capability. So I'd like to acknowledge my team, our funding source, and thank you for your attention.
Thanks for your presentation. We have a couple of minutes for questions from the audience. Questions? I can maybe start with one. So if I understood correctly, essentially your study is mostly based on in silico data, right? So I was wondering what would be a way to actually validate your method on real cases?
Yeah, it's a very good question. I think that I don't need to be too optimistic about it. So we have two separate works in our lab. One is about deep learning with like a lot of cases, and another one is just a validation to the clinical data. But if you validate it to the clinical data, you need manually to check each case, and it's really two separate projects. Right now, it's not possible to integrate the real clinical data into like the deep learning pipeline. And I can say that like it's already some even FDA-approved trial, clinical trial for using cardiac digital twin, but not deep learning for cardiac digital twin. So it's possible. There are some examples, like, and even in clinics you can find some of them, but not merging together, but not the merge.
Right, so you will go for a clinical trial.
Yeah. So this is our plan, but again, I'm not too optimistic, like in terms of like maybe several years about it.
Questions? Yes, it was very clear. Let's thank our speaker again.
It's my pleasure to introduce the second speaker, who will talk about feasibility and benefits of joint learning from MRI databases with different brain diseases and modalities for segmentation, and will be presented by Wentian Xu. You have the floor.
Thanks for the introduction. Hello, everyone. Welcome to see this presentation. I'm Wentian Xu from Konstantinos Kamenitzis Group at the University of Oxford. And this is my first time attend the conference. I'm very exciting. And the topic is feasibility and benefits of joint learning from MRI databases with different brain diseases and modalities for segmentation. First, the brain disease segmentation. Typical methods for brain disease segmentation segment one specific pathology, train on limited pathology-specific database. As a result, they always have low generalizability, and the accuracy is highly dependent on size of databases for the specific pathology. So instead of training the module only on one database, only on one pathology, training on multiple databases or pathologies may be a better choice. Look at these pictures. They are all different brain diseases, and they are quite different from the healthy part and have some similar features. As a result, the general information get from some of the pathologies may help segment other pathology. So we want the model to see different databases with different pathologies and to improve the generalizability and reduce dependence on size of each database. In fact, getting information from different databases are common for working in computer vision. However, for medical images, they have something special, especially for the image type we are working on, the MRI. The MRI, the magnetic resonance imaging, contains different MRI modalities. Like shown in the picture, the tumor database, the brass, have 4 different modalities, and each modality contains different information. You can see some parts of the pathologies are only obvious on some modalities. As a result, we want the model to get features from each modality to improve the segmentation result. Moreover, you can see that for different databases, they may have different sets of modalities. As a result, we want to train our model on multiple MRI databases with different pathology and also with different sets of modalities. Here, we show the 5 databases we use for training. They have different sets of modalities and different pathologies. Labels for all the pathologies are binary marks, and for the 5 databases, 6 modalities are available. Now come to the related work. Some work have been done in the similar area. Some group train their model on multiple databases for lesion detection or structure segmentation. However, they only focus on one modality like CT. Different databases, same modality. And some group reduce degradation when missing some modalities. This means they only use part of the modalities when testing, and they focus on missing modalities and only use one database for training, one database with a set of modalities. And some group focus on transfer learning. They use information from other pathologies to pre-train the model, and however, they only use one modality for each input. You can see in the picture, they may have different databases and different modalities, but for each input, only one modality. In contrast, our work focuses on how to handle diverse training databases with different methodologies and sets of modalities. We train on different databases, different sets of modalities, and for one sample, like shown in the picture, we can have a group of modalities. Now, I want to summarize the target of this work. We want to develop a method that can jointly train on databases with heterogeneous sets of ML modalities and types of brain regions. And we want to explore the benefits of this process in a variety of experiments, including tests on the same databases as training, tests on different sets of modalities, tests on unseen pathologies, and fine-tuning. Now, come to the method. The typical technical challenge for this is to learn on different sets of modalities and predict with diverse sets of modalities. We developed 3 methods, include multi-unite, LF-unite, and MAF-unite. First, I will talk about our main method, the multi-unite. So first, we want to handling diverse modality sites. What we do, we just concatenate all the modalities together. This means each channel presents one modality. Here, we're showing brass as an example, and for other databases, they are similar. And we set the channel size for all the input matrix as the number of all the modalities in available databases. You can see for the 5 databases on the left, the channel size will be 6. And for modalities not available for this sample, we fill them with 0. With that concatenated process, we can train the model with different sets of modalities. However, the model still only sees some specific combination of modalities, and it's difficult to predict with diverse combinations. So what we do, we randomly drop some modalities when training and fill them with 0. Because it's a random job, so even for the sample from the same databases, they may have different sets of modalities. As a result, the model can see diverse combinations when training, and this can improve their generalizability. Now, with these 2 processes and the base unite model, we get the multi-unite method. We also develop more complex architectures like LF-unite and MAF-unite. The LF-unite is inspired by a famous work for missing modalities, the HEMIS. And we use similar architecture for joint training on different sets of modalities. And the MAF-unite is a further extension of LF-unite. We use a multiple encoder and only one decoder for this model. We set some experiments for choosing the architectures. We finally found that multi-unite, the simplest approach, works best. Compared with other methods, multi-unite, the one that concatenate the modalities and use random drop on modalities, has similar or better performance on all the 3 experiments. I will not go into the detail of these experiments because I have to save time for other experiments that show the benefits of joint training. And the multi-unite also need less memory and computation resource. As a result, the multi-unite works best, and we choose multi-unite for all the other experiments. Now, come to these experiments. We set 4 groups of experiments to explore the benefits of joint training on multiple ML databases. Include train and test on multiple ML databases, segment known pathology on sets of modalities different from those used in training, segment pathology never seen during training, and fine-tuning. First, I want to talk about the train-one model on multiple databases with varying modalities. So can it learn all the tasks shown in training? Here, the model are trained on all the 5 databases and test on each database. We can see the result in the table. Compared with the model that trained on only one database, the model trained on multiple databases get comparable or better result. And the results are improved on some small databases like MSPsych and TBI. And we also explore if the model can segment known pathology in new databases with different sets of modalities. Now, the model are still trained on all 5 databases, but the difference now is tested on another stroke database, the SLIS, which is unseen during training. The SLIS have the same pathology as the atlas, the stroke, but different sets of modalities. It have more modalities than atlas, the T2 and FLAIR. We're showing the result here. One just test on the stroke database, the SLIS, with only T1, the model trained on 5 databases perform better than the model trained on Atlas. However, and I want to show something more interesting. However, the model never seen FLAIR and T2 in Atlas. We can see that the model can still use FLAIR and T2 to get better result on Atlas. You can see the changing of the death score in the chart. And this because they see these modalities from other databases, and they can really use this information to improve the result. This is another case that we have new databases with less modalities. The model trained on 5 databases also perform better. Here, we want to go to another experiment, the segment unseen pathologies. Now, the model are trained on 4 out of 5 databases and test on the remaining database. Here we use BRAS as an example. Here's the result. We can see the first 2 rows is the unsupervised method, and 3rd and 4th row is the method trained on other databases, is our method. All these models do not see BRAS when training, and we can see that our method performs better than the unsupervised method, and it can use more modalities to get better result. This means training on other databases really give information to the model to segment on BRAS. And if you have more, and for other databases, they have we got similar result. However, when compared with the supervised method, the upper bound, the model trained on the target database, our methods are still significantly below the supervised method. So we want to know if we have some target data, if we can fine-tune on it. Yeah. We still train the model on 4 out of the 5 databases as a pre-train. However, we fine-tune it on part of the remaining databases. We use a limited number of data for fine-tune. We can, as the result showing, we can see that after fine-tune, the model performs better than the model that only train on the limited number of data. And if we have more data, the fine-tuned model also performs better or comparable than the model that only trained on the target data. And this means that the pre-trained model can still get information from that 4 databases to segment to give information to the new pathologies. Finally, I want to give you some take-home message. So it's wise to try joint training on multiple databases with different pathologies and sets of modalities. So it's possible with simple methods, no complex architectures needed. It can improve performance on small and unseen databases, segment known pathologies on new sets of modalities, and facilitate learning to segment new pathologies with little data. Finally, I want to give a big thanks to all the other people that contribute to this work. Thank you for listening. Any questions?
Thank you very much for the presentation. So we have a couple of minutes for questions. I already see a question here, one of the first couple of rows.
Thank you very much for the presentation. I have a question with respect to the fusion of features. So could you elaborate, please, on the feature, how you performed the fusion of features, and what effect did it have on the performance? And could you explain why?
Sorry, I'm not sure if I heard your question clearly. You mean the feature of the fusion?
No, I mean, can you go to the even before, where you have the architectures?
Oh, yeah?
Where you have, wait. Can you go forward? Where you have models, like 2 models, late fusion and multi-attention. Yeah, this one.
You mean this one, yeah.
So here, if you see the blue point here, fusion in the late fusion unit,
yeah,
and also multi-scale attention fusion unit.
Yeah.
So could you elaborate on the effect of using the first way of fusing the features compared to the second one, and how it affected the performance?
You mean the difference between the first one and the second one?
Yeah.
Like the first one is that we get the feature, we get the embedding after a unit, and then we use fusion method to fuse them together, and then get the segmentation mask. And the second one is different. It's inside the unit. we get the, like we fuse the features on each scape of the unit and also the bottom of the unit, and then we use one decoder to get the result.
Okay, and the second one was better, or like, have you compared the models?
You mean this one, or?
Like the output, like the performance of both these models.
Which performance?
Like database.
Because like.
Okay, maybe I should have a look at the paper. Yeah, thank you.
There's also this poster session. There was a question here up front first. Fourth row.
Yes, thank you for this presentation. I was wondering if you tried to replace the missing modalities with a cycle-GAN, for example, because now it feels you're replacing it with zeros. Maybe it would improve performance. Have you thought about that?
Oh, thanks for your questions. And I think that's maybe a good idea for like future work. But what we want to explore here is to know if that we just use this concatenated way, and if you just use some random job, if it can have some benefits from joint training on this set of modalities. And I think, and also if you like, because here it's not something like missing modalities, if you're just using cycle-GAN, I'm not really sure if you can give more information like to give them the new modalities. If you can't get new information from the cycle-GAN, maybe it's also not improve the result of this work.
Okay, thanks.
There was also, I think, a last question in the back over there.
Hey, did you also, sorry, this way.
Sorry? Oh.
Did you also try the missing modalities in the case of late fusion? Like, did you drop the modalities in case when you're fusing the late fusion network?
You mean the late fusion network?
This one.
We also tried like random job in the late fusion network, and we found that this is also very important for these networks, and we compared it with the original method, both with the random job, but still not perform like better.
I see.
I think in the interest of time we need to continue, so thanks for all your questions, and otherwise try to find the speaker afterwards.
Thank you.
Okay, so we move on to the last but not least talk for this session with title SINER, Spline Enhanced Implicit Neural Representation for Multimodal Registration, will be presented by Vasiliki Sideri-Lampreza. Apologies for my pronunciation.
Impressive. Thank you.
The floor is yours.
Thank you for the kind introduction. So my name is Vasiliki, and I would like to welcome you in our presentation, which is titled Spline Enhanced Implicit Neural Representation for Multimodal Registration. So I'm sure that most of you are familiar with image registration, but just as a short recap, the goal of image registration is to bring two or more images into one common coordinate system by establishing correspondences between them. And this is very applicable in many, it has many applications such as to monitor diseases or, for example, analyze cardiac motion or do population analysis. But let's have a look how this is done in practice. So given a fixed and a moving image, the goal of registration is to find the optimal transformation in the deformable case that minimizes this objective function, which is comprised by two terms. One that is a dissimilarity term between a fixed and a warped moving image, and another term that is a weighted regularizer on the deformation field. So in the iterative optimization case, this is done per image pair, which has the advantage that is more accurate for this pair, but it's also slower if we want to repeat the process many times. And also, the more complex the transformation we want to recover is, the slower the registration becomes. So, as an alternative, deep learning-based methods were introduced where the registration is estimated over a training set, using a training set. And then, while this is very, very slow during training, it's substantially faster during inference because it can perform one-pass inference between a pair of images. However, if we want to infer the transformation at a different resolution, this requires retraining. And also, it is less accurate than pairwise methods. And last but not least, it requires, again, retraining when we want to fine-tune for another modality, for example. And especially when we are dealing with medical data, this can be scarce, so we don't have these large data sets available all the time. So very recently, some years ago, a new way of representing signals was introduced that's called implicit neural representations, which are just simple MLPs that take as input coordinates, and then they represent in their weights and biases the signal. So this is done either using positional embeddings to control how much high and low frequency components can be encoded in the networks, or using periodic activation functions such as sine functions, for example. And these architectures, you can see that the lower part is called siren. And these are coordinate-based representations. They are not tailored to any resolution, which means that we can query them in the resolution that we want. So they offer this pseudo-continuous, they are pseudo-continuous more or less. And last but not least, they are lightweight and they can run in a commodity GPU, which, as we heard from the talk before, it's very important nowadays. So, all right, this is great. This was already done for registration some years ago by Waltering et al. So what they tried to do is to use an INR to represent the deformable transformation between two CT-lang images, which has the advantages of pairwise optimization methods and implicit neural representation methods. And then I said, okay, great, let's try this in my data, which is like inter-subject brain data of the same and different modalities. And then I started running into some issues such as if, for example, the most important and pronounced issue was if I didn't sample the coordinates precisely using, for example, segmentation masks, then the ability of the INR to represent the transformation was very poor. Also, I noticed that for brains, it was very, very prone to folding. That wasn't able to mitigate with just weighting the regularizer higher. And in the multimodal case, the batch size affected the convergence and the speed of the method when using mutual information, because the INR was benefited from smaller batch sizes, whereas the mutual information from higher. And yeah, we ran into some issues there. And in order to circumvent this, we revisited the registration literature. So we revisited specifically free-form deformations, whose idea is that we can manipulate an object using a mesh of control points instead of the object itself. And then this, for image registration, it translates that instead of defining the transformation at each coordinate in space, at each point in space, we can just estimate it in a control point, in the control points, and then use B-spline interpolation to get the dense transformation at the end, which is smoother by construction, and also B-splines are locally controlled, and as a result, they are efficient to calculate when the number of control points is high. So we tried to merge to get the best of both worlds. So we tried to use an INR to represent the transformation, but instead of querying all the coordinates, we decided to incorporate these free-form deformations in the method. So we get a subset of control points, so 1 every 2 or 1 every 4, that results in 1/8 or 1/16 of the whole coordinates, which makes the method to converge faster as well. Then we have this INR with the MLP with sign activation functions to represent the transformation on the control points. And after that, we end up using B-spline interpolation to get the transformation at the end between a pair of images. So as I said, this has certain advantages, such as that we don't need all the coordinates, or we don't need to prioritize which samples we are going to use. And also it's inherently smoother, so we don't have to use a high regularization weight in the end that could affect the convergence of the network. So we try to evaluate our method on a T1 intersubject brain registration and T1 T2 brain registration, and we also evaluate it against several baselines that are like MIRTK, which is a conventional one that supports free-form deformations, also two CNN ones, one with and one without, the previous INR method with ReLU and Siren, yeah, and our method, more or less. And the results were very encouraging, and in terms of accuracy, they were very nice. Otherwise, probably we wouldn't be here. But what I wanted to say is that although we managed to mitigate some of the folding, as you can see on the picture at the bottom and on the quantitative results, there is still room for improvement compared to iterative optimization methods, conventional ones, and learning-based methods. So to sum up, so we like INRs. We find them very appealing because they can register fast in a commodity GPU, and we believe that this can be applicable in many clinical applications as well because of that. And especially for registration, we managed to produce smoother transformation with the incorporation of the FFD, mitigate the need for sampling prioritization, and achieve for both mono and multimodal registration good results. And I also have to say that by using FFD with INRs was just a try, and I think there is still room for improvement and many more things for people to try out. Yeah, and thank you very much, and yeah, happy to take questions.
Thanks. I think we have plenty of time for questions. Yeah, there is one over there. Oh, okay.
Thank you for a question. So maybe we can take a look at the experimental results. Like I'm quite interested in that MIRTK, which is like a classical iterative method, right? So I'm just wondering the method it's using. Like it's using also FFD or like B-spline methods because it's resulting in very low folding folding pixels. So maybe you can elaborate on the comparison or the parameter usage in the MIRTK and in the SINR. Thank you.
Thank you for the question. So if I understand correctly, you are asking if the iterative optimization method uses FFD or not, right? Yes, it does use the FFD. And there was something else about your question, but I'm sorry I didn't get it.
Yeah, thank you. So you mentioned that with the Siren or in the INR, you have like a better, smoother assumption or regularization implicitly embedded in the model. So why is the folding is more compared to MIRTK? So it's
so I think although we managed to lower the folding ratio compared to the original INR, I think it's still prone to folding. And although with the addition of FFD it's smoother, I don't think we managed to match the performance of the conventional method, unfortunately.
Yeah, thank you. I think that answers my question. Thank you.
Hey, thanks for the interesting talk. I think you're using bending energy as a regularization term on top of the implicit regularization provided by the FFD or B-spline. Could you elaborate a little bit on how you try to, or whether you can try to calibrate the bending energy penalty, the weight for it, to get similar folding, or maybe also look at some other metrics of deformation complexity?
Yes, thank you for the question. So at the beginning, this is what we started to do. So we were weighting the bending energy more and more to see if we can actually achieve the result without having to incorporate the FFD. But the more weights that we added in the regularizer, the more it affected the optimization. So at the end, I think the regularizer was affecting the whole process more than the actual loss function. So I couldn't make it, I couldn't stabilize it. And So this is for the networks that use signs, whereas for the ones that use ReLUs, I don't think that you can take the second derivative of the network that uses ReLUs because this is 0. So we had, I think, to use Jacobian regularizers, so first order, but the results were similar, I would say. But I also haven't experimented with just these two.
Thanks. Yeah, there is a question over there.
Thank you for your very nice and clear presentation. If I understood correctly, the use for deep learning registration was that it's very fast, but maybe a bit less accurate. But now you also introduce a pairwise INR that you kind of have to optimize, which I guess also takes maybe quite some time. Could you maybe put that a bit in perspective, how it's then now for the time and performance trade-off?
I had some extra slide for this. So this, for example, how it compares with the methods compare for time. So I think the MIRTK, the comparison with MIRTK, that's conventional method, is not exactly fair because it doesn't support GPU. So of course, then it's going to take longer. But as you see, for example, the INR methods were relatively fast with like 1.5 minutes you could fit them. And yes, like if you train any deep learning algorithm and then you just use it at inference, it's like the performance is achieved in milliseconds. So yeah, this is why I say that the deep learning are still faster, like substantially faster.
Thank you.
You're welcome.
And there was another question next to Joran.
Yes, hello. Did you experiment with less control points? Maybe it improves the folding issue?
Yes, I mean, you have to find a trade-off between like how many control points you are using, depending on how much accuracy you want to achieve. Because, for example, in this case that you want somehow a detailed transformation, the coarser your control point lattice is, then the smoother the transformation, and might not be as accurate. But for example, if you used it for a different purpose, I don't know, like lang registration, then probably you could, or cardiac motion, you could benefit from a different control point spacing. But I think you also have to take into consideration like the spacing of your images, if you have isotropic spacing and all this kind of information that you get from the header to actually tune it.
Okay. One final short burning question. There is one over here.
I also have a question about the control point. Does the control point in your work are located uniformly along the whole image? And what if we set more control points in areas that it may have a more severe deformation?
Mm-hmm. I think there were some works in the past, like 2000s or something, that they did this with conventional methods. This introduces some challenges as well because you have to find clever heuristics how to do this or not. I think it's possible, but we haven't investigated yet. But I think it would be a good idea for future work.
Okay, thank you.
Okay, thanks everyone. Let's thank our speaker again. And I guess this concludes this session, and I think next is a coffee break and then a poster session.
Hi everyone. Welcome to the oral session on weekly and semi-supervised learning. We can welcome Benjamin Billot to start the first presentation. The title is Network Conditioning for Synergetic Learning on Partial Annotation.
Okay. Can start? Okay. So, hi everyone, my name is Benjamin Billot, and I'm a postdoc at MIT. So today I will present my work in collaboration with Boston Children's Hospital on network conditioning to tackle the problem of partial annotations. So overall, the goal here is to perform automated segmentation in 3D. And in general, segmentation networks are trained in a supervised fashion, but this requires a lot of costly labels. So for this reason, the community is studying other types of supervisions, like self, weak, and partial supervision. And today we will focus on partial supervision, where each training image can be labeled for a different subset of all the organs to segment. So this is a very interesting scenario because it enables us either to reduce the annotation cost compared to full supervision, or for the same cost, to train on many more images. So moreover, partial supervision is a common scenario. So for example, let's say that we have a large data set and we want to train a network to automatically segment a specific organ. So out of all these images, we're simply going to manually annotate a few of them. But later, we might be interested in another organ, so again, we manually annotate it, but not necessarily in the same cases. And we see that repeating this process several times naturally leads to partial labels. Moreover, partial supervision also arises in multi-dataset scenarios, where the goal is to combine different supervision from different datasets. So overall, the question here is how can we combine these partial annotations to train a segmentation model? So a first solution, of course, is to train different networks for each individual label, but this is very inefficient in terms of time and resources. So for this reason, we want to train a unified network, which besides efficiency, offers two more advantages, and especially if we look at the training data. Because first, the unified network is trained on much more images than each individual network, but also in the same spirit as multitask learning, training the unified network using several labels enables us to leverage complementary information across these labels such that we improve the representations of the network. The problem with a unified network is that we cannot train it with a classical supervised loss. Indeed, if we look at the partial ground truth, we can see that the missing organs are simply replaced with background, such that if we use a supervised loss, the predictions for these missing organs are simply going to be forced to be zero, which is not correct. So for this reason, a lot of works have proposed special losses for partial supervision, but all these losses assume that labels must be mutually exclusive, which is often not true for partial annotations. Indeed, bear in mind that in the single and multi-dataset scenarios, the organs can be labeled independently such that they can be defined at different levels of details, and thus they can be overlapping. So to deal with those potentially overlapping regions, here we adopt a common strategy known as region-based segmentation, where we train a unified network with sigmoid activations instead of a softmax, such that we can predict binary predictions for each label individually. And in this case, the partial supervision losses that I mentioned earlier all amount to the same thing, which is simply to average sub-losses that were computed independently for each region in the partial ground truth. Okay, so now that we have the general framework for partial supervision, I want to emphasize that several works have tried to improve the results by proposing a new idea, which is simply to specialize parts of the architecture. So for example, by using label-specific encoders, decoders, or last layers. So to summarize, there are two main ideas in the literature. First, we want to train a unified network to model cross-label synergies, but also we want to specialize parts of this network to extract label-specific features. And here we are going to build on these two ideas to propose our new method. So specifically, we propose to shift paradigm and to tackle partial supervision with network conditioning. So as before, we still use a unified network where each block in blue consists of 2 convolutions with ReLU activations. But now we propose to deal with these partial annotations by segmenting one label at a time, which is done by using a label conditioning mechanism that dynamically adapts the features of the network based on the label to segment. So importantly, network conditioning was already proposed for partial supervision, but first it was done with a different conditioning mechanism, and I'm going to come back to this. But also, it was done only for the last layer, so it goes back to this idea of only specializing a fixed part of the architecture. But here, instead of rigidly enforcing which part of the architecture should be specialized, we propose to condition the entire network such that we let it learn where to extract features that are label-specific or that are cross-label information. So one more thing is that here, segmenting labels on demand, like we do, is very useful for the partial supervision scenario because it enables us to retrieve predictions only at the desired level of detail without having to compute the rest of the labels. Okay, so now the question is, how does the conditioning work? So here we use theme layers, which stand for feature-wise linear modulation, and we use them to dynamically adapt the output of every convolutional layers based on the label to segment. So in more detail, here we take every label map and we multiply and add it with 2 parameters, and we do this independently for each feature map, and all those parameters are regrouped in the vectors gamma and beta. And importantly, the value of those vectors directly depend on the label to segment, since they are predicted with an MLP that takes the label as input as a one-hot vector. Also, I want to emphasize that here we use theme layers because they are more stable and faster to train than previous conditioning methods. So for example, the hypernetworks or the conditioning methods of the method I've just talked about earlier, which is a kind of a mix between a hypernetwork and some convolutional layer, but that was very unstable. Okay, so now we can simply complete our framework by inserting theme layers after all convolutions and by using an MLP to predict all the gammas and betas. So in the end, we have achieved our goal of capturing both label-specific information with the MLP as well as cross-label synergies with the shared unit. Finally, let's see how we train this network. So a typical training step starts by randomly selecting an example and performing online data augmentation to improve the robustness of the network. Then we take the image and the label and pass it to the network to obtain a prediction. And finally, we compute the soft dice loss, which is backpropagated to update the weights of the MLP and the shared unit. Okay, so for the results, we evaluated our method in 3 experiments. Here, in terms of data, we use a data set of around 300 fetal scans in 3D that come with partial labels for 5 regions of very different shapes. And then we divide the whole thing between training, validation, and testing. So I just want to emphasize that this fetal data is very challenging to analyze. First, because of the fetal motion that forces to acquire fast sequences with low SNR and resolution, but also because of the high morphological variability that is due to the rapidly evolving fetal anatomy. So in terms of competing methods, we first evaluate individual networks that we then reuse to train a unified network with a pseudo-labeling strategy. Then we include 3 variants of the partial supervision loss with different architectures, so either unified network or label-specific last layer or label-specific decoder. Next, we have DODNet, which is the state-of-the-art in partial supervision by conditioning only the last layer and using also, like I said, a different conditioning mechanism. And of course, we have our method that we name CONEMOS for conditional network for multi-organ segmentation. So in the first experiment, we simply segment each test scan with all methods. And here we start with a visual assessment where we see that our method Conemos in the last column generally obtains better results than the competitors, for which major mistakes are shown with the arrows. So to better quantify those results, we computed die scores as well as the Hausdorff distance for all regions and all methods. And here we see that our method Conemos in green outperforms all the baselines in 9 cases out of 10, and with statistical significance in 6 cases. So in more detail, our method beats the state-of-the-art DODNet in blue by an average of 2 dice points, and is also much more robust because it obtains less outliers. Also, it is more stable because you can see that DODNet for the eyes the results dropped substantially. So we explain these results with the fact that our method is able to capture both label-specific and cross-label information, and it does so in a more flexible and stable way than DODNet, because here we condition the entire network. Okay, so to verify this explanation, here we conducted an ablation where we study the overall accuracy as a function of the number of condition layers. So here we start with the fully conditioned network, and we progressively remove film layers. And in the end, we see that more conditioning leads to a substantial improvement by giving more flexibility and more freedom to the network, and by letting it learn where to specialize its representations. And finally, in the last experiment, we wanted to study the accuracy of all methods when simulating progressively smaller training data sets. And here we see that the gap between our method Conemos in grid and the competitors increases when using fewer training examples, which suggests that our method is more robust to small data sets. And finally, I just want to say that this gain in accuracy and robustness of our method is done while maintaining similar properties in terms of memory requirements, training time, and inference time. So to summarize, we have presented a new method to tackle partial supervision, and this relies on network conditioning to extract label-specific and cross-label information. This was done by conditioning the entire network to let it learn where and how to specialize its representations. Then we obtain state-of-the-art results while maintaining a good compromise between accuracy, memory requirements, training time, and inference time. And also, we are more robust to small data sets, which is very important when trying to reduce the overall annotation effort. And finally, future work will first look at how to segment new organs without retraining our method from scratch, and how to integrate it with semi- and self-supervised learning methods to be able to leverage completely unlabeled data. So I would like to thank my collaborators, and that's it for me. Thank you very much for listening, and I'm happy to take questions.
There is some question here.
Thanks. Thanks for a very nice talk. So if I understand correctly, you're doing this in a multi-label setting where the labels are independent and not necessarily mutually exclusive, but sometimes they would be. So how do you handle, for example, resolving the boundary between mutually exclusive regions?
So that's the whole thing, like you're not forced to. So that's like I said, like you can here retrieve predictions at different levels of details, and whatever detail of whatever level of details interests you the most, that's the one you're going to go for. So that's like here you can play on several coarse or fine grid of labels, which is not possible when you predict all the labels at the same time. So you have this flexibility here.
But sometimes there would be, you would want to predict 2 neighboring tissues, right? So you would have to resolve this.
Yeah, sure. And then, so I'm not saying like this is exactly the same as a unified network that would solve this problem for you. So then maybe you can do some post-processing. But yeah, I mean, the flexibility comes at the cost of like sometimes having to solve this problem. Yeah.
Thanks.
But my question was, you have now this very nice model, and
sorry, can you speak a bit louder?
Can you hear me better now? Okay.
Yes.
Yes. So thank you for the nice talk. you have this very nice model. In order to predict now multiple labels at the same time, it's not possible, right?
No, it's not possible. So you need to do like iterative segmentation. So here, if I can go back. So we predict 5 labels, and here you see that the overall time is very competitive with the other method. It's still like sub-second predictions. But then, of course, like this difference would grow if you start labeling more structures. So like, for example, if you want to segment 50, like, yes, there will be a significant gap. But then, like, if you want to segment 200 labels, maybe you don't need all of them. So here, our method gives you the flexibility to choose whichever you want to segment. So yeah, like again, like this is the flexibility comes at some price, but then those are different things.
Thank you. I have a second question. Have you had a look at the embeddings that you feed into your model, how they change over the layers?
Yes.
This could be very interesting. I guess they're more important toward the end.
Yeah, so I didn't visualize the features per se, but I would love to. I mean, I didn't have time enough to finish the paper to include this. But I think that, the ablation study here shows you that like basically the way the network learns how to like specialize its representations is all along the network, throughout the network. So it's progressive. There's no like, it doesn't learn that like all representations should be specialized only at the end or only at the beginning. It's quite diffuse across the whole architecture.
Okay, and in this plot, it's starting from the end towards the start, right?
Exactly. I start removing the theme layers at the start.
Thank you for your very nice talk. I was wondering how many of those layers are shared across all the convolutional blocks, and how many of the parameters are then distinct. And
so there is a different theme layer for every convolution. But at the end of the day, it's not a lot of parameters. So it's like the number of output features per convolution times 2 because you have a gamma and a beta. So this is really ridiculous in terms of memory requirements and number of parameters. Like I showed here, like, yeah, it's nearly the same as like a regular unit. So it doesn't add too much.
Thank you very much.
Thank you for the very nice talk. I was wondering, is it really different from conditioning on text input? When you have multiple tokens with attention that can modulate your features. What is commonly found in generators like Stable Diffusion, when you can condition the output of your generator based on some.
So to be clear here, it's not conditioned on text, but on one hot vector representation of the labels.
Yeah.
I'm not sure why you would use text in this context.
No,
here.
but you could replace words with just multiple vectors, so that instead of having one vector that is conditioning everything, you could have multiple vectors. Like you could ask only one inference, but for several classes.
Ah, okay, I see what you mean. Yeah. So you could start playing with this, but then I think that like in terms of memory requirements, you would explode the whole thing. And also in terms of training time. So yeah, I mean, I didn't go for this, but also it would be interesting to try.
Okay, thanks.
I guess we can go to the next presentation. Thank you again.
Okay, let's move to the next presentation. Meanwhile, there are seats in the front for people who are standing in the back. So the next paper is from Barlow twins to triplet training, differentiating dementia with limited data, and Yitong Li from Technical University of Munich is going to present.
Thank you very much for the introduction. Hello everyone, my name is Yitong, and I'm from the Lab for AI in Medical Imaging in Technical University of Munich. So I'm very happy today to present our work from Barlow twins to triplet training. We want to differentiate different types of dementia with limited data. So dementia is like an umbrella term that can describe different types of cognitive impairment, including Alzheimer's disease, frontotemporal dementia, vascular dementia, and Lewy body dementia. And particularly in our work, we want to differentiate 2 types of dementia, Alzheimer's disease and frontotemporal dementia. As for Alzheimer's disease is already a widely explored domain, and a lot of work has been focusing on how to diagnose Alzheimer's patients from the healthy subjects. However, we want to focus on the differential diagnosis, which is more clinical valuable, especially for these two types of dementia, because first, they are the two most common dementia types among early onset patients, which means patients that are under 65 years old. And secondly, they have very similar clinical symptoms, so it's very difficult for doctors to differentiate these two types in the hospital, because for the other two types, they can have more clear pathological symptoms that you can test through like blood tests and other types of techniques. And also, they too can have mutually exclusive treatment. For example, one inhibitors widely used for the treatment of Alzheimer's disease can be poisonous for the frontotemporal dementia patients. And to differentiate these two types, you can focus on the brain atrophy patterns on the brain structures. Because like for Alzheimer's disease, the atrophy mainly focuses on the parietal lobe and temporal lobe, but for the frontotemporal dementia, as the name infers, the atrophy mainly focuses on the frontal lobe and temporal lobe. So we would like to use the 3D MRI to differentiate these two, because MRI can give us very clear atrophy patterns. And also, given the very good performance of deep neural network during the recent years, we also want to leverage deep neural network to extract the features end-to-end for the diagnosis. However, there's a very large obstacles when we want to use deep neural network in medical imaging domain, which is we always have very limited availability of medical data. It's mostly because of privacy issue and ethical issue, and sometimes there are some diseases that are just very rare to be collected. For example, in our local hospital, over the several years, we can only collect like 76 subjects for the frontotemporal dementia. So this is an amount that is far lower for a good training of deep neural network, because you can easily face issues like overfitting. But luckily, thanks to the big medical imaging community, we now have large public data sets, for example, like any data sets, but they only include control and Alzheimer's disease, and an FD dataset, which include control and FTD cases. And we call it a task-related dataset. And also, there's a far larger dataset called UK Biobank, which includes almost 40,000 images. However, they are mostly unlabeled subjects and mostly healthy subjects there. So we call it a task-unrelated dataset. And some people may want to combine this ANI and NFD dataset, so you can have a larger data for training. However, we have to be aware of the danger of confounding issues by simply combining two different datasets, because the classifiers may just differentiate different types of datasets instead of the diagnostic labels. So we want to introduce a methodology that can leverage these large datasets, but on the other hand, we want to overcome these confounding issues and also overfitting issues. So here comes our overall introduction of our methodology. We have 3 steps. So first, we want to pre-train a large self-supervised learning model on the large task-unrelated dataset, and then some knowledge distillation on the task-related dataset, and finally, after we get this good embedding model, we can fine-tune it on our in-house dataset. So let's go into step by step. So first, for self-supervised learning, on the task-unlabeled dataset, there are several methods, and we choose contrastive learning method here. So for contrastive learning method, it's very intuitive. So we have one set of input image, and we have 2 crops by applying 2 different types of data augmentation there. And then after feature extractor and the projector, we have 2 sets of features. So for different methods, it have different objective to deal with these 2 different sets of features. For example, for SimClear, it wants the crops from the same image to be closer together, but the crops from different images to be far away from each other. And in the end, we choose Barlow twins to be our self-supervised learning method. It has several benefits. One of the benefits is that it's very less sensitive to the batch size, because as we know, for the self-supervised learning method, it always requires you to have a very large batch size. But we are using 3D volume, so it will be really hard to train if we want to use a large batch size. And Barlow twins, so it's similar as what I said before. It's also, the main core is the contrastive learning method. But after we get 2 batches of different crops, we want the correlation matrix between these two sets of features to be an identity matrix. And then, after we pre-train on the large task unrelated datasets, we use the pre-trained encoder as the teacher network and then freeze it. Then we randomly initialize the student network here. So during the self-distillation, we want first the features from the teacher network and the student network to be minimized of the KL divergence. And then we also want to train the student network to our related task, so the cross-entropy loss of the student's prediction between the ground truth labels are also minimized. So you may ask, like, why don't you just use a normal fine-tuning on the task-related dataset? We also did that, but empirically it gave us worse results. Potentially, we hypothesize it's because it will produce some overfitting towards the task-related dataset. But once we include this KL divergence here, it functions like a regulation term so that it helps to still preserve the knowledge we learned from the previous step. And then, in the end, finally, we can fine-tune it on our task-specific data, so we do a normal differential diagnosis task in the end. So overall, we introduce our triplet training, so it includes 3 steps. First, the self-supervised learning on the task-unrelated large dataset, then self-distillation on the task-related dataset, and finally, fine-tuning on your downstream task. So now let's see some results. So we always report the differential diagnosis on our in-house dataset, which is a single-site dataset, after 5-fold cross-validation. So first, we want to compare what if we just do a normal supervised learning on our in-house small dataset. Of course, it doesn't give us very good results, like only 67% of balanced accuracy, which is probably because of overfitting. And then we want to first do a supervised training on the task-related dataset because it also has labels, and then fine-tune it on our in-house dataset. But still, you only get 1% of improvement. And then we want to include this large task-unrelated dataset UK Biobank by just first apply a state-of-the-art self-supervised learning methods, and then we fine-tune it on our specific data. So surprisingly, we can see if we use Barlow twins, like BT here, we can already get a very high performance improvement here. So it means it's very beneficial to include even large task-unrelated data here to help with your downstream task. And finally, in the end, if we include all 3 steps of our triplet training, it gives us much better results, and we can get 75% of balanced accuracy in the end. So we hypothesize that triplet training may potentially mitigate overfitting when training with limited data. And we also want to further interpret the underlying reason behind it by visualizing the latent space with UMAP after each step of triplet training. So we visualize it on both all the datasets as in the first row and also some stepwise target datasets. So we use these purple dots for the large unlabeled UK Biobank data, and these greenish dots for the control subjects, the reddish dots for Alzheimer's patients, and grayish dots for the frontotemporal dementia patients. So we can see if we just, after the first step, just a self-supervised learning method, all the data mixing together, which is understandable because the self-supervised learning doesn't have any task-related training there. And then, if we introduce the self-distillation on the task-related dataset, we can already see 3 clear clusters here. However, we can see these clusters are mainly on the task-related data because we are training on top of it. However, it's still not generalizable enough because you can see on the unseen dataset, that is our in-house dataset, these dots are still mixing together everywhere. It's not clustered yet. And then finally, after the final step of triplet training, you can already see 3 very distinct clusters divided by the diagnostic labels, and it's regardless of the datasets. So first, we can see this network didn't unlearn the previous knowledge while fitting on the new domain. And also, the high accuracy of triplet training is probably rooted in because the decision boundaries of the classifier is less population dependent, because we can see the clusters, they are not divided by the datasets, but by the diagnostic labels in the end. And also we want to see what if we use different types of self-supervised learning method in the initial step, like other state-of-the-art, SimClear, VCRIDE, or DIRA. And we can see the triplet training can always give you high and consistent accuracy across all different methods. So it highlights the generalizability and robustness of our framework. So in the end, as conclusion, we introduced a new framework called triplet training. So we want to take advantage of large task unrelated or related data sets so to help with our downstream task. And they show high robustness and generalizability in the end. And also it's worth noting that we do always report the differential diagnosis our consistent single-site clinical data set. And so I'm sorry I forgot to put the group photo here in the end. You can scan the QR code to see our group faces. Okay, thank you very much for your attention.
Thank you. Any questions? While people think of their questions, I will ask one. If I understood well, I was sitting a little bit on the side, but if I understood well from your plot, you're claiming a significant advantage when you actually fine-tune on the in-house dataset instead of just doing this on the ADNI, right?
Yeah, because in the end, we want to do differential diagnosis, but it's always hard to find the data that include all these data sets, different types.
So I'm trying to see here. Okay, so you are, the fine-tuning actually helps a lot. What is the accuracy in this accuracy
75%.
When you're doing the fine-tuning?
Yeah, after fine-tuning, it's 75%.
Then my question is, you had a relatively small data set.
Yeah.
Did you actually try with even less data just to get an idea on, how low can you go if you do this kind of tricks?
Yeah, actually we did because at first the hospital only gave us in total like 72 subjects.
Mm-hmm.
So in the beginning, we only have our in-house data set with 72 subjects. But actually we get a good performance, like 86%, but after 5-4 course validation. Okay. But also we get a bit high variance because the data is become even less, and so the variance can be high, and when you do the fine-tuning, it can be unstable.
No, but I mean, did you try fine-tuning with only, say, half of the 72 subjects to see if you can still do it?
Yeah, exactly, like 60% of the 72, yeah.
Okay. Okay, question?
Yes, hello, thank you for the presentation. So one question, if you come back to the slide with the self-supervised methods.
Yeah.
I think that was one of the last ones.
Yeah.
I mean the table that you show.
Okay.
No, I meant this other table, sorry.
The other table?
Yes, exactly.
Okay.
So what are the differences between these methods?
Yeah, so basically they are SimClear, VCRIDE, and Balo twins. They are all contrastive learning methods. They have very similar core idea, but they just differentiate difference in the objectives. For example, Balo twins, they want the correlation matrix to be identity, and SimClear is just the info NCE loss. And VCRIDE is on top of the Balo twins, so it's very similar as Balo twins, but it then includes one more loss, that is the variance loss. It wants the standard deviation inside the one batch to be higher than one threshold. So this method all try to prevent the mode collapse. Very often it happened in self-supervised learning method. And DIRA is a method introduced in CVPR 2022. So in addition to the contrastive learning, it also includes reconstructive loss and also an adversarial loss. So it's like a method. Oh, I actually have a more detailed structure of these different methods.
Oh.
So yeah, so you can see like this is all trying to do similar task, but have a bit more different losses there.
Okay, thank you so much for the overview.
Okay, no problem.
Hello. Hey, thank you for the. It's here.
Yeah.
Hey.
Hello.
Yeah. Thank you for the presentation. And I was curious about the zero-shot properties of this model, because after you fine-tune it on the task-related data set, have you tried it? Have you tried like zero-shotting it on the in-house data set?
After fine-tuning on the data sets?
After fine-tuning on the related data set. So you had this 3 steps, right?
Yeah.
Task-related, you fine-tune it on the, you do the self-distillation on the, yeah, this one. You do the self-distillation on the task-related data set, right?
Yeah.
And I was just curious, what was like the zero-shot properties if you tried any on the in-house data set? So without further fine-tuning, how does the model perform in the zero-shot setting?
Yeah, actually, if you just do a zero-shot without fine-tuning, the results are not so good. You can also infer from the cluster here, like after self-distillation, the dots from our in-house data set is still wandering around. It may be because of the data like domain adaptation issue, because still after you train down the task-related data, they are not in the same distribution as our in-house data set. So it's still very important to do the fine-tuning in the end.
Okay, thank you.
Yeah.
Can I ask a last question? Okay. Do you do anything so that your network doesn't forget what it learned during the pre-training or in the self-distillation step? Or do you just train in each step, I don't know, until convergence?
So it's trained sequentially. So first you do the pre-training and then do it one step by step. I'm sorry, is that your question?
Partially, I would say. So do you do anything specific to keep it close to what it learned during each step?
So for example, in the self-distillation, we have this KL divergence. So it tries to preserve the knowledge from the previous step. And then in the in-house data set, there's no specific technique here. You just do a normal fine-tuning there. And then it will give you the results.
Okay, so if there are no more questions, let's thank the speaker and move on to the next talk.
Thank you.
Now we'll hear Finn Berendt for his work on combining reconstruction based on supervised anomaly detection with supervised segmentation for brain MRI.
Yes. Thank you. And welcome to this presentation. I'm Finn Berendt from... Okay. I hope you can hear me now. Okay, it sounds better now. So again, I'm Finn Berendt from the Hamburg University of Technology. And together with my colleagues, we've been working on unsupervised anomaly detection, and more precisely, we proposed a framework to combine reconstruction-based anomaly detection with supervised methods. And before I go into the details of anomaly detection or our framework, I want to quickly introduce unsupervised anomaly detection for those who don't know. The overall idea is to detect and segment all kinds of anomalies. I can even try to get it a bit higher. And instead of supervised methods where we would, for example, use the BRADS data set to train a UNet, which will result in a very precise model in segmenting tumors, the approach in unsupervised anomaly detection is a bit more general, because if you would now use a stroke lesion for this tumor-trained model, it might fail. So therefore, there are different approaches to this UAD, unsupervised anomaly detection, that are aimed to be more general for any kind of anomalies. And one would be a reconstruction-based approach, which is very prominent in brain MRIs. And here, in the first step, a generative model is trained based on healthy training data to reconstruct healthy training data. And the theory is that if we have a trained model that reconstructs healthy brain MRIs and we input an unhealthy model, and I symbolize this with a star, an unhealthy image, the generative model will fail to reconstruct the abnormal patterns in the image, and it will estimate some sort of pseudo-healthy reconstruction out of the abnormal image. And now if you look at the two images, you could use the difference between input and reconstruction to get an anomaly map or anomaly score. And so, as I said, this is very prominent in brain MRI. However, there's another approach which is prominent recently, or this approach is not new, but in brain MRI there are some works that work on synthetic training data. So here the idea is to first generate unhealthy images with an anomaly generator and to add a synthetic anomaly into a healthy brain image. And since we added the synthetic anomaly ourselves, we now know where it is and we have a label. So we can train a segmentation network to get an anomaly score, and we use a supervised loss to train the segmentation network. And I want to quickly compare these two methods. And I have two scenarios. One would be the known pathology scenario, where we train our model on a known pathology and evaluate it on the same. And here you can see that with a supervised approach, the segmentation network, we can get very precise segmentations. While for the reconstruction-based approach, while we can get a broad idea where the anomaly is, there are lots of false positives or blurry regions in this anomaly map that come due to imperfect reconstructions of the model, because the model might also fail to reconstruct the little details in the healthy parts of the image. On the other hand, if we have an unknown pathology, which means the supervised network has not seen this anomaly before, the supervised network might also fail to get this anomaly or pathology, while the reconstruction-based method still has its false positives here, but it is a more general solution, and you can get a broad idea where the anomaly is still. So therefore, our idea was to get a method that gets both the enhanced precision of segmentation models, or supervised segmentation models, and the generalizability of UAD methods. So our framework is called SIDM, stands for Supervised Anomaly Detection with Diffusion Models. And even though it's called with diffusion models, you could use any kind of generative model here. So in the first stage, we train on healthy data. And we train the generative model to reconstruct these healthy data samples, just as in the UAD case, the reconstruction-based case. In the second stage, we use an anomaly generator and generate abnormal images ourselves with known locations now. And now instead of feeding this abnormal image directly to the segmentation network, we take our generative model, freeze the weights, and get our pseudo-healthy reconstruction given the unhealthy image. And now use the discrepancy of input and reconstruction as input to the segmentation network. And the segmentation network is then trained based on our label that we have. So therefore, we aim to learn to predict the anomaly or pathology given the deviation of input and reconstruction. And at test time, we assume that we have an abnormal image right now, and we again use our generative model to reconstruct a pseudo-healthy reconstruction, or to generate a pseudo-healthy reconstruction, and use the difference to feed it into the segmentation network. Now, we could use this score directly for anomaly prediction. However, still it might have a strong prior toward the anomalies that are seen during training. And therefore, we add another path, which we call the unsupervised branch. So the upper branch would be the segmentation branch now, and the lower path would be the reconstruction branch or unsupervised branch. And here we use the SSAM, a metric to compare two images, in a pixel-wise fashion to get an additional anomaly score, which is more general. So now we combine these two anomaly scores by simple addition. And the idea is that the segmentation branch will be very good for known pathologies. While if we have an unknown pathology, our reconstruction-based branch will be very general. So therefore, this framework aims to get best of both worlds, so to say. And we evaluate this based in different settings. So the first training stage is always the same. We use the IXI dataset with healthy training data to train our generative model. Now, in the second stage, we have different approaches. One would be the self-supervised approach, because synthetic training data is also often called self-supervised. And we use the DREM method for generating anomalies. And the DREM method comes from industrial defect detection, so it's not very close to real anomalies or pathologies, but we want to show that this can even work with unreal pathologies or anomalies. And in the second setting, we use the weakly supervised setting, where we have small subsets of our real pathologies from the BRADS dataset, which contain tumors, and the ATLAS datasets, which contains stroke lesions. And we evaluate our models on all different datasets. So, coming to the results, the first question we had was, okay, which generative model should we use? As I said, you could use any kind of generative model. However, we compared some unsupervised baselines, and we found out, or our results show, that conditioned GDPMs perform quite well for real pathologies, and that's why we chose CDDPMs as a baseline here. For the segmentation network, we use a basic UNet. So, coming to the results for self-supervision. Here you can see the baseline CDDPM performance, which is the unsupervised performance, no labeled data at all. Then, and our goal would now be to beat this performance for known pathologies and maintain this performance for unknown pathologies. So, here you can see the results if we train our network and the baseline UNet with the DREM anomalies. So here we use synthetic anomalies from the DREM method, and you can see that the UNet performs worse compared to the unsupervised baseline, which shows, or which indicates that the used anomalies are not very close to the real pathologies here. However, our SIDM method seems to maintain the performance or even slightly improves the performance of the unsupervised method. And if we now look at the weakly supervised setting, if we train our method with a BRADS dataset, or a small part of a BRADS dataset, meaning using tumors, and evaluating on tumors, we can see performance improvements for both the baseline UNet, which, by the way, only takes in the abnormal input and predicts the segmentation map. And if we compare this to the ATLAS dataset, so we are training with tumors and evaluating on stroke lesions, the UNet does not perform very good here. However, our SIDM method is able to maintain this general performance even though the pathology is unknown. And the same applies if we train with the stroke dataset, the ATLAS dataset, where we can improve the performance for the known pathology while maintaining the general performance of the UAD method. So, coming to a quick discussion and conclusion, we have shown that our proposed framework can enhance the generalization when using synthetic anomalies. And as I said, these synthetic anomalies are not really close to real pathologies. However, our method is still able to get something out of this data, which is not the case for the basic UNet. And furthermore, we can use a small subset of labeled data without losing the generalization capabilities of unsupervised methods. And this is very important because in UAD we often say we only have, or we do not have any labeled data, which is not true actually, because there are some labeled data sets out there, and we have shown a method that could make use of these data sets for no cost, or at least not decreasing the performance for other data sets. And what we want to do further is we want to see if our method works with different data sets, maybe even different modalities, and especially with more realistic anomalies. Because, as I said, they are not very realistic, and there's a lot of work going on in designing these types of anomalies to be more realistic. And furthermore, we want to see if our method or our framework could actually improve the segmentation performance of a very efficient segmentation model that is trained on the full BRADS dataset, for example. So, but now it's time to end my talk. I hope you enjoyed it, and also, for pictures of us, you have to go to our institute website. Yes, and if you have questions now, you can ask them, or maybe catch me at the conference. Thank you.
Thanks for your talk. One question
Hi.
here.
I have a question.
Mhm.
So I missed it maybe, but how do you generate images with anomalies from images without anomalies?
Well, basically, we took an existing method to do this. So the DREM method adds specific patterns or damages to the images, and this will result in an abnormal image with a region where the
But you
abnormality
say
is.
this is not realistic.
Well, the gradients, for example, are not smooth in these images, which is not the case for real pathologies, for example, so therefore it's not really realistic and might be easy to detect or to overfit on.
And can you say a word how DREM is working?
Sorry?
Can you explain in a very short way how DREM, this method, is working? Is it a bit like data augmentation, or is it like smarter?
As far as I remember, they use some sort of noise, Perlin noise, and they binarize this noise map given some threshold, and then you would have like a map of coarse blobs and maybe stripes, and given these map, they add some intensity changes or I don't know every type of augmentation by heart right now, but you can look it up in either the appendix of our paper or in the original paper. I hope that helps.
So, thank you very much. You said that you assume that each image at test time has an anomaly. Does your method start to hallucinate contents or into images that don't have an anomaly?
I hope I understand correctly. You're asking if we include healthy images during evaluation? Okay. Basically, we do not include healthy volumes. But since we evaluate slice-wise, we do have some slices without anomalies.
Okay, thank you.
Next question, please.
Hi, thank you for the talk. Sorry, I'm here.
Okay.
Hi, sorry. Just a question about the approach in general. I was just wondering how sensitive it is to the size of the lesion you're trying to detect or the anomaly, like, or maybe if your method is less sensitive to, like.
Well, the unsupervised branch is quite sensitive to the size of a lesion. That's what I can say. The supervised branch might be less sensitive depending on the data you use to train it.
Okay, thank you.
Oh, I'm sorry, we need to move on to the next talk. Please thank the speaker again.
Okay. So the final talk is IHCS Corgan, an unsupervised generative adversarial network for end-to-end Ki-67 scoring for clinical breast cancer diagnosis. And Carl Morler is going to do the presentation.
Hello, thank you so much for joining today. The topic of my presentation, I won't say it again. My name is Carl Molnar. I'm the first author of this paper. Our group is affiliated with the Mayo Clinic. Highlighted below is the contributors to this paper. We're a multidisciplinary team of computational pathologists, computer scientists, and pathologists. Let's start with a little bit of background. The image on the right is an immunohistochemistry stain, or IHC stain. Specifically, we've applied a Ki-67 protein biomarker. This protein biomarker indicates the rate of cancer proliferation in cells, and this information is useful for pathologists in order to aid in patient prognosis and guide treatment. So what pathologists would like to do is count the ratio of all the brown cells in this image versus the total cells in this image, and then given this percentage, they can come to a clinical diagnosis. Typically, this process is performed manually by pathologists. However, this is very error-prone and laborious. it'd be extremely desirable to have a very robust automated K67 scoring solution in order to save time and potentially reduce human error. In the literature, these approaches are typically supervised, meaning someone must manually annotate all of the cells in an image on the order of hundreds of thousands, and they would pass a K67 stain tile along with its manual annotation into something like an autoencoder, such as UNet, which is very commonly used for medical image analysis. We also look at DeepLif as a non-autoencoder option. Instead of using manual annotations, DeepLif uses a complex co-registered staining process along with the K67 stain tile. It would perform some multiplex immunofluorescence staining, and by algorithmically combining these, they can produce an annotation that is automatic. However, it's very time-consuming, and it requires some specialized and specific equipment that costs millions of dollars, to my understanding. The problem with these supervised methods is that you need to train them on a data set, and since it takes so much data to train, you would hope that the supervised model is very robust. But one of the problems with this is that if you have a data set that the model is trained on, which is very different from the data that you're trying to inference with, then you'll run into many problems with your supervised model. So, as you can see in the top row, this is a public data set called the BC Data dataset. The IEC stain images have a very different background from our internal data set. It's much more yellow. But aside from that, there are other issues with this data set. If you look very closely, you can see some cell texture differences. There is some white space around our region of interest. And further, there's some dark stroma staining in our images, which is not present in the public data set. So this is going to lead to many problems if we try to use a supervised model, which is trained on an out-of-distribution data set. Wouldn't it be very useful to have a model which could be trained without needing all of these manual annotations? So this leads us to looking at an unsupervised paradigm. Before we talk about some unsupervised methods, I'd like to just briefly go over the foundation of some of these methods. This is shown is a traditional GAN architecture. How these would work is typically you would have a source domain image. I've shown it as real X. This would be domain X, and you try to generate an image from domain Y. In a traditional GAN, you're trying to fool the discriminator into thinking that the generated image is actually a real image. However, the problem with the traditional GAN is that you haven't forced the model to produce any image in the target domain which corresponds to the input domain. So many research has been looking at a cycle GAN, and cycle GAN works like this. They train a generator in the reverse direction, so they would take an image from domain X, and then they would generate an image on domain Y, and then they would generate back onto domain X. And this forces the model to learn a specific and unique mapping onto domain Y such that the input can be reconstructed. And for completeness, I've added the equations. And also I've added this reverse direction. And the purpose of this reverse direction mapping from the target domain onto the source domain is to let the generator F see some real data as it's training. So this creates a more robust reverse generator. So how can this be used in practice? Well, if we take an IHC image and we were to transfer it into the H&E domain, then possibly we could produce a segmentation of the IHC images because we can pass the generated H&E images through a pre-trained H&E segmentation model. And this would be not quite a K67 scoring, but it would at least get us the segmentation of the IHC image, which might help a little bit with pathologist counting of the cells. However, what we've seen here is that this is now a two-stage process. We've taken an IHC image, then we have to turn it into an H&E image, and then we have to pass it through a pre-trained H&E segmentation model, such as HoverNet. So if we go a step further, what if we first applied the pre-trained H&E segmentation model to the H&E directly, and then we pass that as a target input? So that's what this research does. It shows that we can actually learn the morphology from an H&E H&E stain and apply this directly to the morphology of the IHC. And this actually works quite well. And it's a one-stage process. However, still, we haven't learned information that we can use for scoring because we didn't retain the type of the cells. We didn't retain which cells are blue and which cells are brown. And further, we don't actually have a count of cells. We only have a binary segmentation. But this leads us into our research. Our method, we take an H&E segmentation and then we randomly color the cells as green or red. Further, we apply a random shade to these cells in order to capture the differences in intensity values in the source IHC image. Actually, this works very well. Our model can learn this correspondence between a brown cell and a red color in the target synthetic data set, and a blue cell and a green color in the target synthetic data set. And the reason that it can learn this is because, again, the cycle loss, it needs to generate an image in the target domain such that the source image can be reconstructed. And the only way that it can retain the color information is by using the color channels in the target data set. However, we still don't have a Ki-67 scoring. We just have a lot of blobs. So what we do is we extract further information from the H&E images, including the center point distance from each cell and its center point, and then we extract a red and a green binary segmentation mask, which is based on the random colors that we've applied to the H&E cells. And the model learns this alongside the main branch. And from this information, we can just apply a simple local maxima algorithm on the cell center points to extract the cell instances, and then apply an argmax operation on the cell instances to extract the predicted cell types. One further detail, the main branch actually, the encoder X to the decoder Y shown in the image, it's learning all the information needed in order to generate data for the fake K. So we can detach the gradients going from the decoder K to the encoder X. And this is a type of task decomposition that helps improve the model's stability. So to dig in a little bit more about our model's inputs, like we've seen before, our input database is just a tiled region of interest from our in-house IHC database, along with a case-level clinical diagnosis. And our target data set is extracted from the TCGA's breast carcinoma project. These are all H&E images. So we selected 23 slides from this database, we tiled them, and then we passed them through a pre-trained H&E segmentation model. From that, we get tiled cell centers and contours. We randomly color these cell contours, and then we extract the two norm center point distances and red and green binary segmentation masks. And these are the input to our model. During inferencing, it's a very simple process. We simply take an IHC image, we pass it through the model's encoder and the secondary decoder, and we get the cell center points, distance maps, and the red and green binary segmentation masks. We apply a local maxima algorithm again on the cell center points, and then we apply an argmax on the predicted cell types. Then we get a Ki-67 tile scoring. We can aggregate all of these per region of interest in order to create a predicted case report. And then we compare this against the ground truth case report coming from the pathologists. Typically, these are thresholded by a 20% cutoff. So if a case has over 20% Ki-67 positive cells, then it's classified as overall Ki-67 positive. And this indicates whether pathologists should give chemotherapy, for example, to a breast cancer patient. So on to experiments. Our internal data set features 2,126 Ki-67 whole slide images with annotated region of interest. We extracted Ki-67 scores directly from pathology reports, and then we used a 20% classification cutoff based on the clinical guidelines. And then we can evaluate a binary classification by measuring precision recall accuracy and F1 score. The target synthetic data set has been extracted from 23 H&E stain images, sourced from TCA's breast carcinoma project, and then we randomly select 10,000 target data set tiles. So we evaluated 2 experiments in our internal data set. The first is a random train test split. So we randomly pick out cases from our data set, and then we evaluate against supervised models UNet and DeepLift, which have been trained on an out-of-domain data set. Our model performs significantly better. Further, we apply a chronological train test split. So we train our models on cases collected before 2018, and then we test on cases collected after 2018. Actually, the supervised models perform even worse on this data, and the reason for this is a chronological data drift. On the more recent cases, we noticed that the stroma staining was much darker, and it threw off the supervised models much more. We also performed a head-to-head comparison on a public data set called the BC Data dataset, which features 1,338 K67 stain images. We can evaluate against the mean average error of cells in these images. So we separate the mean average error of positive cells and the mean average error of negative cells per image. And then we use the same target synthetic data set to train our model. So the results on the public data set, we noticed that our model, when trained on the public data set, was just a little bit worse than the best supervised model. However, we didn't use the annotations in order to train our model. Shown is a visualization of some of the results. In magenta is the supervised model's error. In yellow is our model's error, and in green is both of our model's error. And shown in blue is the negative predictions, and in red is the positive predictions. I'd like to specifically point out the second column in this, which indicates some regions where the stroma actually threw off the supervised model because it's much darker in this image. Finally, in conclusion, our model shows promise in a clinical setting where it performed much better than the supervised model trained on out-of-domain data. It also offers an alternative to supervised scoring methods, which need a lot of manual annotations in order to train. The applications of this, the applications of our work include situations where ground truth manual annotations are not possible, and also it tackles the problem of data drift. If your data is constantly shifting, it might be better to train an unsupervised model on this data, because otherwise you'll have to collect all of the labels again. It could also serve as a starting point for new projects where you're not sure if an unsupervised model might be sufficient. In that case, you might as well try our model, and it may perform quite well. And that's it for my presentation.
Okay, thank you.
Thank you.
So this is the last talk of the day, so let's see if we have any questions. Questions? Oh, there's one here.
Hi. I want to ask you you only talked about when the manual annotations are not available. Let's say we have some of them available. Do you have some thoughts of improving the accuracy in that case, like incorporating some annotations?
Well, if you would have some manual annotations available, you might want to try something like fine-tuning the supervised model. So maybe you would train it on out-of-distribution data and then use it to fine-tune a supervised model. So that may be an option. But in terms of having some annotations for our model, it would not help.
Okay, thank you.
Yes, thank you.
To page 13, page 13. And And I wonder how to use the three-channel fake K to generate the KI 67 tile scoring, as I know the second size is the maybe blue cell and the third is the red cell. So what is the use of the first size of the three?
So we want to make sure you were asking how do I generate the scoring from the fake K?
Maybe the use why the use of the distance to central in your work, the first size.
I'm sorry, I can't.
Maybe can you turn to the main figure?
Oh yes, this one.
Yeah.
Yes.
What's the use of the center point distance map in this work? Can you just use the red cell binary mask and the green cell binary mask?
Yeah, so the reason that I've added this fake K is because if we only generate fake Y, we still don't have a K67 scoring. So you might be able to apply an algorithm such as watershed to get an instance segmentation, but you would still have to threshold the cell types, and watershed is not that good at separating overlapping cells. So that led us to go the direction of a fake K, which is actually trying to predict instances of cells by predicting the center point distances. So the center point distances allow us to apply a local maxima algorithm in order to extract the cell instances. And then once we have the cell instances, we can do a binary segmentation, or we can do an argmax over the binary segmentation masks. And the reason that we've added these binary segmentation masks is specifically because in our real Y, we've added different shades to the green and the red cells. And so this is not conducive to cell type prediction. So if we do a binary segmentation, then it's a very strong prediction of whether or not there's a cell there. However, if we're just looking at the shadings, you might get a very small predictive value.
Another question is about, have you computed the dice between the predicted binary mask and the ground truth binary mask?
Can you say one more time? I'm so sorry.
The dice score between the predicted.
A dice score between the predictive segmentation mask and which one? I'm sorry.
Do you have ground truth mask in the dataset?
We don't have any ground truth segmentation masks in our data set.
You just use the pre-trained model of the HNE?
Oh, yes. But we haven't applied the pre-trained H&E segmentation to IHC, so we don't have any ground truth segmentation of our IHC images.
Okay, I know. Thank you.
Thank you.
Okay. Any more questions?
Yep, I'll keep it short. So thank you very much for your interesting presentation. And I was kind of wondering, in like a view of clinical applicability, do you know what the inter-observer variability is of different pathologists that look at these same slides? Is that comparable to what you guys are doing, or are they still a step above?
Yeah, this is an interesting question. It's not something that we've specifically looked at, but it's something that can lead to human error not only in the manual annotation of cells, which is used for the supervised models, but it's also something that can lead to differences in the case reports that we used. So this is very important, and is something that we do not directly address in this work, but it's definitely worth further study.
Okay, thank you.
Okay, and I think
is it okay? Okay. So I just have a quick question. Thank you for the great presentation. I wonder, like, this work can be like extended to the other, to figure out other kind of IHC images status. For example, like HR2 status or the PR positive, ER positive, something like that. I know that those kind of IHC image have different kind of scoring system or something.
Yes.
So yeah, just want to hear about your opinion.
Yeah, this is an excellent question, and we're also thinking about how this model might be applied to something like an ER-PR stain, which there are different levels of brown stains which you must factor in. We have not specifically done this study yet. However, we're thinking perhaps you can do a manual threshold of the fake Y, which actually is predicting the intensity value of the brown, and this might be used to predict whether a cell is 1+, 2+, 3+, etc., and is very important for the pathologists.
Okay, thank you.
Okay, let's thank the speaker.
Okay, and now to finish the afternoon session, we'll have a short sponsor session. It's an academic one, it's not an industrial one, and so we welcome a research network from the southeast part of France. It's called Prime, and Michael Zika will present his activity.
Thanks. Thank you. First, don't worry. I know this is the last talk of the day, so I won't be long. We are a consortium of 22 teams from 15 labs in France, and we get funding from the French government to work on physics, radiobiology, imaging, and simulation. We do that by providing funding for students, do scientific animation, such as organizing or sponsoring a research day, conference, and a workshop. And we do have also some action in education and training. The main scientific themes where we work, our main clinical application are cancer and aging disease. Our works can be centered on the detection, the detector and the sensor for this application, but also the image acquisition, simulation for radiotherapy, and also biology, pure biology, and with the images produced with all these work package. We also do works on image analysis and of course with deep learning and learning. Few work to present ourselves. In these works, it's for radiotherapy. We need to do a simulation of SPECT images. Here, the objective was to reduce the computation burden. Simulation is traditionally done with Monte Carlo simulation, which is very heavy, time-consuming. Here, the Monte Carlo simulation is replaced by a GAN. So we model the path of the photon inside the body of a patient. So the input of the network is a noise vector because it's a stochastic process. The network is conditioned on the position and direction and the energy of the photon when it enters the body, and we try to estimate the same measure, but when it goes outside the body. So as you can see, the results are pretty similar between the Monte Carlo experiment and the GAN-based results, but the computation is 100 times faster. In this second work, it's try to when we work on 2D images, on cardiac images, the images themselves may not be consistent temporarily because of the reconstruction and the synchronization. This implies that when you try to segment the images, the segmentation are not consistent temporally. So in this work, the space of the segmentation are encoded with an ARVAE, trying to fit some meaningful variable in the latent space. And we see that when we smooth temporarily this latent space, when we do have two effects, first the segmentation are more consistent visually. We do reduce the Hausdorff metric error, and this opened the way to define a biomarker based on temporality. The last work that I present to you is about enhancing the interpretability of deep networks. So we have the case of a simple classifier, for example, for diagnosis. So we want to discriminate between healthy and pathological data, for example, tumor data. When we work on using a post-hoc attribution method at the relevance map, well, we expect to see something that might be centered on the pathology, at least what we would like to get, but this is not always the case. So in this work, we did use post-hoc method during the training with this simple constraint that when it's a healthy subject that is at the input of the network, the attribution map shouldn't point any part of the image, so it should be completely blue. And we noticed that using this unsupervised constraint, well, this revealed the tumor of the MS lesion on the pathological data. So we do have also some action on education and training. For example, we offer an open facility for training in imaging. So if you do have teaching to do in imaging and you want to provide a hand-on session to your student, but you don't have the material to do that, for example, in X-ray, ultrasound, MRI, and optics, we do have some facility that are open to people outside our labs. For now, it's only usable in place, but next year there will be remote access to certain of the hands-on, so you will be able to use the facility wherever in the world. If you do want some more information, please contact Thomas and Bruno that are here. And my last slide is about the DLMI summer school that we organized with people in ETS in Montreal and University of Sherbrooke. So it's a summer school that we organize every year. This year it's in Montreal, it's next week. And next year it will be in France, in Lyon, and we shift every year. So there is every subject from the very basics of deep learning to very advanced topics such as a foundation model and weak supervision. And thank you.
So thank you. We have room for a very short question because we need to leave the room before, like in 5 minutes. So if you have some question. So then we close the session for today. Thank you again.